MSc work that's going on. We have GMP facility, non-human primate model and primate center, and we have expertise in canine and feline models. And so we leveraged all our expertise. And so why MSCs? I think the biggest advantage of using MSCs is you can use allogenic MSCs for transplantation or transfer, because you don't need to have autologous. That way you avoid, if any, there are defects in those cells in those individuals, then you're not using the same ones and putting them back. And so allogenic transplantation works. It has anti-inflammatory molecules that it secretes, so homes to the site of inflammation. In animal models, it's been very well tested. And giving MSCs in mouse model increased vaccine responses. And I think until 2023, I think almost more than 1,000 clinical trials have been registered or what has been done. Most of the work is not in infectious disease area, but in HIV area, there have been safety studies done, and they seem to be safe. In COVID, they have been tried for the viral infection, but most of them were in really advanced end stage. And so we then performed this study where SIV-infected animals at 4 weeks when gut epithelium is disrupted and the CD4 T cells are depleted, microenvironment is impaired, and at 4 weeks we gave 3 infusions every 2 weeks, and then at 10 weeks we examined the tissue. And these are bone marrow-derived allogenic MSCs that were given. And what we found was very surprising. These are the animals, there is no antiretroviral therapy or nothing, just MSCs given to the virus, the virally infected animals. And we saw that, as you see in blue color, the viral loads decreased in the peripheral blood, and CD4 T cells in peripheral blood, as you can see in blue color, got stabilized. And so we asked, what is the reason and what's happening in the gut? And you can see in the left, SIV-infected animals, virus RNA positive cells are spread all over in the villi of the gut, the finger-like projections, effector sites in the gut. But if you look at the right-hand side, where MSC-treated animals, they are completely cleared. Obviously, all those cells got killed or removed, and a lot of virus got trapped in the germinal centers in lymphoid follicles. So we tried to understand the clearance, why did that occur, and we could clearly see that virus-specific CD8 T cell responses increased, as you see in the blue bar here, the interferon gamma producing cells. And how much time do I have? 5 minutes? Okay. And then antibodies increased. So what we did then is to look at the tissue and found that actually the germinal centers, as you can see on the top panels, the last picture you can see with the white color, those are all B cells, and the germinal center was very nicely repaired and restored. And the molecular analysis proved that actually B cell proliferation took place. And so what we now see is that the MSCs were able to actually repair the lymphoid follicles. And so the question then we asked is, again, it's a so complicated, complex structure. And so what are the driving mechanisms? And so for that, we needed to have a good, very look overall and use the spatial transcriptomics and proteomics to map the repair pathways in the different sections, and use the GeoMix nanostring platform, which is now with the Bruker. And added on the tissue sections, the RNA probes, which are about 18,000, over 18,000 probes on one slide, and then did also antibody panel, about 85 antibody panel, to map the repair pathways and identify key driving mechanisms for the repair. And here you can see that left side, SIB positive, all disrupted, dysregulated the lymphoid follicles and depleted loss of cells in yellow color, but with MSCs, that all got restored and repaired. And so what we did then is to capture all these regions and just specifically targeted the proteomic and RNA transcriptomic analysis of those regions in B follicle, B cell follicles, germinal center, and in the villi. And clearly there were differences what the gene expression popped up, cytotoxic activity in villi, and B cell regeneration and lymphoid follicle in left-hand side. Okay, and clearly showed the germinal center formation and T follicular helper cells were repaired. So our question then was that if this is happening, even in the absence of ART, what happens? Can MSCs help if you therapy interrupt? Because that is the big question. So with ART, there was complete nice suppression of the virus. We interrupted therapy. The viral loads went up, as you can see, resurgence of the virus. But in MSC-treated animals, as you see in blue, there was massive upsurge of antiviral antibodies and cytotoxic T cell activity. And with that, we then looked at the tissue sections, and again, in the therapy interruption, which is ATI, with MSCs, there was a protection and preservation of the germinal centers and lymphoid follicles. And you can see at the bottom the gut lining, and you can again see there was a protection of the gut lining. So MSCs actually, even in the therapy interruption, intervened and supported and maintained the mucosal functional structures. So overall, MSCs can be used adjunct to ART for enhancing mucosal repair, renewal, and immunity, and leverage this for advanced combination therapy to control the virus and maybe bring it to the level of long-term non-progressor status. And so these are all the different people, wonderful individuals who worked in our group over these projects and the funding. And thank you.
Thank you, Professor Satya. A good round of applause. Thank you, Professor Satya, for delivering a very good and futuristic looking forward for MSC therapies along with ART. And let's see how fast it goes to the market and how fast it really helps the patients. We are not having the questions right now because we're running late. Thank you very much. And it's time to invite the second speaker. I would rather like to stand and introduce the second speaker, Professor Suman Chakravarti. I come from industry, and for me it's really a great honor to listen to somebody who has been at the core of innovations which see the light of the day and translates ideas into production so that it goes into the industry and the patient use. Welcome, Professor Suman Chakravarti. I don't think so I would like to go into his scientific details. Professor Chakravarti comes from Indian Institute of Technology, Kharagpur, and has enormous number of publications, citations, innovations. And let's listen to him more than I talk about his introduction. Welcome, sir.
Thanks very much, and it's a great honor and also a bit of embarrassment to speak in front of some audience who know about a subject and I do not know about a subject. Like immunology, I only know how to spell it. That's the maximum that is my knowledge. Having said that, what I realize is that sometimes lack of knowledge is also not bad, because then we can explore certain things, and that exploration can give rise to some outcome which may be used by the domain experts. So this is I would say one such example or set of examples where we tried to innovate certain technologies for diagnostics, and these technologies are a bit generic in a sense that they could be applied to many different types of disease detection examples. Now, I will like to give a very brief perspective because of the interest of time. I will not get into the exhaustive details. But overall, in my own experience in working in the IIT system now for quite some time, almost close to 25 years, what I have realized is that there is a significant gap in many cases in terms of what research is being done in the lab and what is actually needed on the field. And there are many reasons why these gaps do exist, but usually there is not much drive to reduce this gap or bridge this gap. When I first started working in the domain of these kinds of technologies, I mostly thought about technologies which we develop in the lab and on which we work in the lab. But then I realized, and the location of Kharagpur is one of the what I would say catalysts to get such a thought process, because outside IIT you get complete rural ambience, and then you realize that whatever fancy technologies with which we do work in the lab, those kinds of technologies are not affordable and accessible to people who need these the most. And with that, and possibly, if you see, this is the main building of IIT Kharagpur, and it is written dedicated to the service of the nation. So, I try to answer this question that doing service to the nation is a very big thing, but even if we are not able to do service to our own surrounding community, what we are doing at IITs, and that somehow it is not moving, I do not know.
IT team, please support.
This is not moving.
Someone from IT team?
Okay. So now, one aspect that I would like to emphasize here, because many times we talk about affordable technologies, and to a large extent, affordable technologies are converted to cheap technologies, and those are not really good in the medical domain where accuracy and so many other factors are important. Beyond affordability, one of the major concerns is accessibility, and this is a more subtle concern, because people, even if they can afford, they may not have accessible system where in the need of the hour they can get some intervention. Now, fortunately, over the past several years, more than one decade, there has been significant boom in the digital technology space and telecommunication and internet connectivity. With that, the digital health centers, which were previously not so much active, have become more and more active. And most of these centers are blessed with good telemedicine facilities, and that makes it quite convenient that there is a patient who interfaces with a remote doctor. The remote doctor only has to give some time to understand the language and dialect in which the patient is communicating. Now, typically in the rural scenario, it happens not just by simple telecommunication, but also there is a frontline health worker, typically a minimally trained boy or girl, who will interface with the patient, gather some data which is either in the form of some basic body vital measurement, some symptoms, medical history, whatever is available, and enter that in the software which in a tab or smartphone interface can be visible. Now, with that as an available data, what the remote doctor will do, even if the doctor was a physical doctor sitting in the front, for most of the common issues, it in the first level would become watch and go with some essential medication. So there would not be very high level of decisive recommendation unless it is so much apparent that people have understood it. Just like fever, people will typically see with 3 days with paracetamol whether it is going away, and then in parallel looking for other symptoms which are developing. To that extent, this system is very good because the medicine shops, thanks to the Pradhan Mantri Yojana and several other initiatives, at very low cost you can get generic medicine, and that can be given to these people. But what creates a major problem is when the doctor prescribes a diagnostic test. So the possibilities are that either the person physically has to be taken to a center or the sample has to be taken to a center. If it is medical imaging, then the person physically has to be taken. There is no way out. Otherwise, it is the sample which may be taken. But there are so many issues associated, some technical, the sample degradation may occur, some bit ergonomic, economic, etc. Someone in a poor health condition has to travel, and then the person can become even more sick. The person can lose a daily wage because of that. So many things can happen. So keeping that in view, it would be great if there is an alternative mechanism for diagnostic tests. Again, it is not moving.
Is there someone to help?
Just go to the next slide, that's it. Okay. So, the problem that came up from this very, very common rural healthcare delivery model is diagnostics at resource limited settings. Now, as technology developers, people make a very common mistake, and in my early career I also have made many such mistakes. So the mistake is that we develop a technology in the lab, it comes to a prototype, it is tested and all, and then we think that we will make a product which will come to the use. But conversion of that prototype to product is not straightforward, because the boundary conditions in the lab and that in the field are completely different. So in the lab you have high quality power, you have highly controlled environment, you have reagent, storage, supply chain, skilled technicians. These are quite obvious in the lab. In fact, very senior teachers many times call some students who are working in the lab as useless. Now they will be like gods or goddesses if you compare them with the kinds of people who work in the field and have to do. So if you are having a pipette based sample dispensing system, it may not actually work in the field. So that means that the entire design from the very conception of the technology has to be oriented to its end use, and better it is done, more successful this thing is. But there is always an optimization, because if you want to create a technology which can be used by anybody under any condition, there has to be a lot of automation and control in the technology, and then it will be more expensive. So how can you make a trade-off between these? Typically in an Indian context where getting human resources for such things is not difficult at all. So that is one of the things. So whoever is having control on the computer, if you could just click on the green place, yes. So this is one of the technologies with which we started working, maybe 16, 17 years back, I exactly cannot remember when. So this is generically a colorimetric test for some blood parameter. Let us say plasma glucose, very common test. So what is being done is, so you have a paper strip, we are using something known as paper microfluidics. So you prick the finger with a lancet, the blood is soaked directly into the paper, there is no such pipette or anything. There is a micro channel which is connected with that blood collection spot, and there is a membrane filter which separates the plasma from the whole blood. We actually do a direct plasma based measurement and not a correlation from the whole blood. So the plasma gets separated and the plasma moves by capillary action to the reaction spot where the glucose oxidase peroxidase based standard enzymatic reaction will take place. What we commonly do is we use a side reagent which will generate a color. For example, we use potassium iodide which will generate iodine and it will be a reddish brown color. Now the question is, you have to somehow get that color digitized and extract the quantitative glucose out of that. It requires the imaging and its analysis. There are two ways of doing it. One is you can use the smartphone camera under a controlled lighting condition, and you see this black box. Inside this there is a LED light which has controlled illumination, and the entire thing is microcontroller enabled. So once you have that strip in which the reaction is going on, you insert the strip into the box just like a credit card inserted into a reader, and then the smartphone camera or the camera integrated in the device, they will take the image, and there is a correlation that is developed between the gold standard value and the image features. Long time back, when the AI and machine learning was not that common, that correlation was just like a simple mathematical function. Now we have made it AI based, where we extract the features from the image and train the algorithm to act according to the image, so that it is a dynamic machine learning that is used for such purposes. Now there are quite a few related innovations on which we worked. One is, so what happens here is that you have the plasma separated and the plasma moves from the source where the sample is dispensed to the reaction spot where there are reagents. So it moves by capillary action. Now capillary action is a bit slow process. It has an advantage because you don't require the pump to move the fluid. So you can have a completely pumpless system, which in a resource limited setting is always very good. But the movement is slow. So to make up for the movement, one can use electric field to make the movement faster. Now the challenge that we initially faced is that to make the electric field working across two electrodes, you have to make electrodes. And electrode deposition on such a fine capillary driven system is actually quite a elaborate process, electrode deposition process. So what we try to do is to use a very simple frugal approach, that is the black region that you see is actually sketched by pencil, HB pencil, and it is the graphite which is there in the tip of the pencil, it goes and gets deposited there, and the graphite acts as an electrode. So in that way, without much intention from our research lab, we invented a technology called as paper and pencil microfluidics, which around the world people started using from then onwards. Now of course, when you are making this on a mass scale, you are not using pencil. So you can use the graphite deposition, automated graphite deposition on the paper strip. Now if you go to the next slide, one of the major things See, what I am trying to talk about is a set of platform technologies. That means these technologies, I cannot say that the previous one can be used only for detecting sugar. It could be for several other analytes where the biochemistry or microbiology, these things are different. But the technology platform is the same. There are many such technologies in the labs where we need very powerful microscope, and that many times is a challenge. Typically, if you are doing a fluorescence based analysis, so the fluorescence spectroscopy, it requires also a spectrophotometer. So these things are all expensive things, and although you can make tabletop experiments, the overall cost and infrastructure is not that frugal. So what we tried to do is to develop a lateral flow immunoassay where the antigen antibody binding what is taking place, there is a carbon dot which is attached to the antibody. Now when there is antigen antibody binding and there is an excitation with the ultraviolet light, that binded antigen antibody complex will give rise to emission from based on that excitation, and that emission can be in specific colors depending on specific reactions. So it is possible to actually get a very high quality emission even for trace amount of biomarkers, and this is very important for early screening of diseases. There are many inflammatory biomarkers, and we were working on certain such CA125 and several other inflammatory biomarkers, and these biomarkers are at certain stages at very low concentration, which even the advanced lab technologies also find difficult to detect. But in this way you can have a highly sensitive and specific detection, but what you have to crack for is the alternative for the microscope. So what we did is, by organizing simple objective eyepieces etc., we organized a homemade microscope which in the Indian rupees, if you talk about the entire thing including 3D printed parts, it is 1500 to 2000 rupees, and that microscope can give about 1000x magnification, and that microscope is put in the form of an attachment on that 3D printed part, on the top of which you have the smartphone. So the entire thing is smartphone enabled, and whatever image you get from here, that image, although it is not of that much quality at which a very elaborate microscope and spectrometer system will give, but one can use AI to train these images to work well. Now I am coming to another type of diagnostics which is of course related but a bit different, is the molecular diagnostics. And this kind of work we started during the COVID era, when RT-PCR test became almost like a household name, and every day people were talking about some new RT-PCR kits are being developed, and it actually democratized the system very well. So if some of you could click the top right, so the video will run, top right part of the slide. Yes, that one, top, not this one, the top one. Yes, this one, yes. So what we realized is that it is actually not the kit which is the most important thing, because the real-time PCR machine, having it somewhere and maintaining it with the help of the infrastructure, technicians etc., is like sustaining an elephant. So that is not that straightforward. So what we did is, we tried to use the lamp based technology, which is isothermal nucleic acid amplification, but more importantly we developed a machine where end to end the controlled thermal processes could take place. So first, say for COVID as an example, the nasopharyngeal or oropharyngeal swab is collected, then it is being put into a buffer, and the buffer is heated, and that mixture is heated at 85 degrees centigrade or 85 to 90 roughly for about 2 to 3 minutes. So it will now inactivate the particular solution, and then it is subjected to the gene based reaction, like very similar to RT-PCR, N gene, E gene, S gene, etc. So then what we did is that all those gene based tests, they were all done in this small box where we were having this automated temperature time control. There is no external influence, and after the reaction is over, which is typically 65 degrees centigrade for about 30 minutes. The products which are developed, if you are using a colorimetric lamp, it will give a color, and we were not taking the products out of the system, and that is where the major problem of carryover contamination which is there in molecular diagnostics, that can be addressed. So then again taking the smartphone picture, doing the analysis and correlating the color with the positivity or negativity. Even we have tried to correlate the brightness, etc., different features of the color with the CT value, if it was a RT-PCR test done. So what we realized is that this is a genetic platform technology. After COVID, we have a ICMR Center of Excellence at IIT Kharagpur, and we started working on the MTB the diagnostics. So we are the basic TB diagnostics is done. Now we are looking for the drug resistant, multiple drug resistant strains in the same platform. So, but there are different variants of these kind of infectious disease testing on which we have worked. So one is like antibiotic resistance or antimicrobial resistance. So using a paper strip, how we have done it is, I have given a simple cartoon. So you have folded paper strips, and this is for the UTI where we want to understand that whether there is E. coli infection with significant drug resistance, and it many times happens with hospital assisted UTIs. So what we try to do is to, after isolating the bacteria, we subjected them to certain antibiotics, one drop each on one of these wells, and the papers are folded. So bacterial cultured solution will then react with the antibiotic solution, and then whatever is the outcome, then that folded paper is being brought to that blue colored chemical, that is like chromogen, and if the antibiotic is killing the bacteria, within 3 to 4 hours that entire blue will become pink. So a doctor can keep this small kit at the office, take a small cultured sample and try out as many possible genetic drugs as possible. Before giving an antibiotic, we will actually know which antibiotic might or might not work for such patient. Then we have also been working on saline gargle samples collected from the throat, typically keeping in view that the flu season, where children in the schools are very severely affected and they easily transmit this kind of infection from one to the other, and it is not quite convenient that we do the PCR and all those things. So if you go to the next one, next slide please. Let me see. Okay. So now I am coming towards the end of my presentation, and a couple of more such technologies I will talk about. Now every technology we try to improve upon what we have learned from the previous, because in the world, at least in the technology world, there is nothing called as perfect, and that perfect never comes. So when I was thinking more about the shortcomings of the technology that we have developed, what came to my mind is this box. It is not about the cost of the box, whatever may be the cost. Say there are many patients who throughout the day may have to test their blood sugar. So will they just bring out this box from their bag and start doing in a meeting or somewhere in the airport sitting or in the flight sitting? It is not convenient. Now why is this box required? It is required because it creates a controlled environment where that image is taken and analyzed, because you do not have a control lab, so you require a control. Now someone may ask that I have a smartphone, I can take the picture, what is the problem? Now every smartphone has a different camera setting and background illumination is different. the user will have the difference. So at that time we transferred this technology to a startup company known as Smart QR, and it's about a couple of years back we incubated them at IIT Kharagpur, and they started working on it. So smart means smartphone based and QR means there is a QR coded strip. There is no device. So it's a complete device free technology. You can use your own smartphone and keep the paper strips in your pocket. So this is a hemoglobin test which is known as HemoQR, and similarly, so you see that it is very simple. You collect the thing with the finger prick with the lancet, and then you take the image, and within 1 minute the app will tell what is the hemoglobin concentration. So this is a HemoQR technology. So if you go to the one below and just click it, the blue colored thing, PrEP QR. So this is also very interesting. This is for the testing of vaginal pH by women in a self-collected manner. So although pH collection appears to be obvious, but with at least 1 decimal accuracy, if we cannot decode that vaginal pH with several different types of vaginal infections. So this 1 decimal accurate thing which women can, at least downtrodden women can look into some aspects of their own health and not dependent on many others. This is a final example that we developed a thermal imaging based technology for oral cancer screening, and it's actually not directly thermal imaging. There is a thermal camera which converts the thermal image to a blood perfusion data, set of blood perfusion data, and blood perfusion data is correlated with typically the patient data on pre-cancer, that is not squamous cell carcinoma in this particular example field study that we did with one of the hospitals, Guru Nanak Institute of Dental Science and Research, or it could be even anything else but not cancer. So in that way we used a classification pre-cancer, cancer, and neither of these two, and developed this technology. But then we realized that this is just, we can use it for screening. So we tried to develop an app where a lot of interrogation, self-imaging, etc. can be done and uploaded, and in that way you can get a quantitative screening. On the top of that you use this device. So this kind of technology where part of the interrogation what the doctor does can be entered into the form of a logic, and some essential screening tests, they can be combined to give a clinical decision support. And this is where the future of healthcare technology for limited resource settings is going, where of course you should not substitute a doctor, but what one can do is always get such clinical decision support through technology and let the doctor then have quantitative evidence before coming to an answer. So now these kinds of technologies we worked at, we have been working not primarily in the lab but primarily in the field. So if you could run the two movies, you could see, just the left, the top one and the bottom. The top one is in a rural setting, which is in a place which is district Birbhum, West Bengal. So if you just click the top left, it is just, you can see that in a village how we do this. And the bottom one is that in Sundarbans we use this women's screening technology, and we have launch in which things move. So in a way I would say that it's a bit of a holistic approach for the solution, where it's not just about technology, but it is technology combined with a socio-economic model which creates a type of a different stream of healthcare delivery for resource limited settings, where technology is not just used for the sake of technology, but technology combined with AI and human interference, trying to address many problems for which direct affordability and accessibility is missing. Thank you very much for your attention.
Thank you, Dr. Chakrabarti, for a very interesting talk, and how the well-designed engineering can really help in patient care by on-spot, on-clinic, on-field, on-community diagnostics by reaching them to all these immediate spot analysis. Very interesting talk. There must have been many, many questions, but I think time, does it permit any more question? Perhaps not. During the tea, there can be discussions.
Thank you very
I
much.
thank my co-chair. I thank both the speakers. It is really very heartening to listen to both of you. And thank you, the organizers, to have given us the opportunity to chair this very interesting session. Thanks my co-chair to help me to do the job. And
little more responsibility of handing over the tokens to our speakers. May I request Ma'am Satya to be on the stage again?
Yes. May I lead this session of memento presentation? I would like to request our respected chairperson, Professor Nibriti Das and Dr. Sanjeev Gupta, to kindly present our esteemed speakers with the mementos. May I invite Professor Suman Chakrabarti on the center of the stage to come forward for the felicitation. I would also request our speakers and chairperson to kindly remain on the stage for a group photograph. Followed by him, I would like to invite Professor Satya Dandekar to kindly come forward for the stage for the felicitation, followed. These mementos are a gesture of our heartfelt appreciation for the invaluable insight she has shared with us today. Thank you very much, sir and ma'am, for doing the honors. Now I would like to invite Dr. Amber Kumar Rai, sir, on the stage. Dr. Amber Kumar Rai, the convener of IMMUNOCON, will kindly felicitate the chairpersons of this plenary talk for their insightful words and the encouragement. I would like to apologize from the behalf of MNNIT for the ill working of the pointer, and thanks for our speaker for cooperating. Now, as we move forward, we have the technical segment, a segment that we all await. It is my pleasure to invite the representative of Den-Her Corporation to kindly come forward and lead the session. Den-Her Corporation is a global leader in diagnostic systems and life sciences research technology. Let me give the stage to him. Please welcome him with a huge round of applause.
Hello.
You can either simply ask them to change the slide, or you can use the pointer. If
Okay.
there's any working, you can
Got it.
on and off.
Thank you. So first of all, thank you, the IMMUNOCON team, for helping us coming over the stage and deliver our talk. We have a short time of about 15 minutes. We are from Den-Her Corporation. It's a US-based company, and all the companies listed out there, starting from Cytiva, Abcam, Aldevron, Beckman Coulter, IDT, and many others, probably you might be using in your daily routine, and they were all part of Den-Her Corporation as a group. So we are here to present today from 3 companies. One is Abcam, the other one is Beckman Coulter, and the third one is Molecular Devices. So we will share the dais, and after me, my colleague will come and present for Beckman as well. So moving forward, yeah. Okay, got it. So why choose AppCam? So AppCam is one of the leader for antibodies, and in the biotechnology sector, anybody who is working in immunology, cell biology, or molecular biology or IHC would probably know this name. And why? Why do we want to choose AppCam is that we are recognized by Site IB for the 10th consecutive year for providing high quality antibodies. We almost 50% of the research papers which have antibodies as a part of it are used from AppCam. And also we have about 90,000 antibodies, about 25,000 ELISA kits, and we have about 1,20,000 products in total from AppCam, which are being served to every customer out here in the room as well as otherwise. So that is how we are. And I'm Abhishek Chauhan. You can contact me at the bottom, abhishek.chauhan@appcam.com for anything if you need from AppCam. And we also are there at booth number 3 in the hall there. Moving forward, I think the pointer has some issue. Yeah, okay. Okay, so AppCam is globally known. We have about 2,000 employees all around the world. We are serving at 12 global locations, participating in about more than 150 countries. As I mentioned, 50% of the global research papers are regarding antibodies are having AppCam name. We have more than 25 years of antibody experience, and we have about, as I said, 1.2 lakh products. So that is how we are. We are always, recognized by producing good quality antibody and high quality products from the Site AB company, who are actually representing the antibody market all around the globe. And that is how we are getting awards almost every year for the last 10 years. And these were just 2 representation of the last 2 years awards, which we have from 2024 and 2025. This is our complete product range. Probably many times people are still aware that they know that AppCam is known for only producing antibodies, but we have a wide range of products as well, which have majorly antibodies, about 90,000. And then we have a lot of bioactive proteins, which are at a level of 95% purity and can be used in a research. And then we have got biochemical and cell-based assays, about 18,000 kits from the molecular diagnostic side. Not literally diagnostic, but in the terms research, but molecular-based. And, oops, sorry. And then we have got simple step ELISA, which is a 90-minute ELISA, along with the normal ELISA plates as well. We have conjugation kits, which are used for conjugating or labeling your antibodies. This is a 30-second protocol, very effective product, which we have, and very, very, very well used in all the academic and research segments. We have got some cell engineering products, not popular in India, but we have. As I mentioned, the specialized products from AppCam are antibodies, and out of that, the most important product portfolio is recombinant antibodies. We have more than 32,000 recombinant antibodies, which are high performing. We have 13,000 carrier-free antibodies as well. We have about 8,000 conjugated antibodies, which are already labeled and used for you as a ready-to-use product. And we have got 420 multiclonal antibodies, which behave both as polyclonal as well as monoclonals. And we have got some 190 chimeras. Further on, our Immunotherapy portfolio. This is basically the ELISA kits, which we use in the lab. We have about 28,000. We have about 28,000 kits in total. We have also modified our ELISA kits into a 90-minute protocol. So in the recent times, what we have done at an advanced stage is that all our ELISA kits, about 10% of the total list, can be modified and used as a 90-minute protocol, saving a lot of the time in the research, and that is what we are trying to help you with. So if you see on the top, they have a 90-minute ELISA kits, which are 4 times faster, save a lot of time on your ELISA experiments in the lab. We also have antibody pairs if you want to buy individually. That is also an important component of the lab. And then we have got some protein standards, along with markers and normal label dyes as well. As we mentioned, in total, in the research segment, if you consider the number of ELISA kits which we sell, we save about 70 years of ELISA time in the customer place. This is a research done by us itself, and that saves a lot of time while you do ELISA. We have pre-coated plates. That's the protocol I'm just trying to show here. We have got pre-coated plates, which saves you about 2.5 hours while you do ELISA, and then you add your label antibody and go forward and complete your experiment. We are specialized in oncology segment. We have 19,000 primary antibodies, about 5,000 proteins and peptides. 2,100 cell lines which are ready to use off the shelf. I mean, you can order it directly from us. They are actually knockout cell lines and they are very, very useful. We have got, I mentioned 9,000 to 1,000 alive and antibody pairs, and we have got a lot of biochemical active compounds. For neuroscience, we have 15,000 primary antibodies for about 4,000 proteins and peptides, similarly 15,000 cell lines and lysates. This is a very important slide. I would like to take about a minute on this. So we have got 3 segments in AppChem. So any product which you buy from AppChem, it generally comes under a 1-year warranty. So when you use the product, if it doesn't work well, you can reach out to us and probably we'll try to help you out in whatever particular way. Further on, if you use the product and you want to review the product on our website and give a feedback on how the product is performing along with the pictures and data, we give you AppChem review points. Those points can be also redeemed back in terms of discount while you buy AppChem antibody. The second one, AppChem trial program, it's also very critical because many times you want to see some antibody which might be like a anti-rabbit and you might be using into anti-goat or something like in your lab, and we might not have a research paper to that. If you use that antibody in an anti-goat platform and probably coming out with some result, whether it works for you or if it doesn't work for you, in both the situations, we will give you the same value of the product free of cost back again if you submit all the results to us, whether it is working for you or if it doesn't work for you. So both ways, we are still trying to help the research and probably get the data back and work on our side to make it better for you. The third thing, as I mentioned, every product which you buy from AppChem have 1-year warranty. So whatever you buy yesterday, today, and tomorrow, that will all come under the warranty, and you can always reach out to us for any challenges from AppChem. This is my last slide. As I mentioned here, probably this is smaller, you might not be able to read, but we have products which are recombinant antibodies, simple step ELISA, antibody pairs, and can be used in all the segments, right from academia to the biopharma, which is basic research, target ID validation, compound screening, lead optimization, preclinical development, clinical trials, biomarker discovery and validation, and diagnostic development platform. So this is a very crisp, because we had a very limited time, so I'm just keeping it very short and straightforward. I will ask my other colleague from Beckman Coulter to continue the presentation as such. Thank you very much for attention.
Thank you, Abhishek. Good afternoon, everyone. My name is Jagtar Singh, and I'm from Beckman Coulter. I will take you through the journey of innovation in flow cytometer from Beckman Coulter. I'll skip this slide because this is the intellectual property, which is a disclaimer. So we have a pedigree of history in innovation in flow cytometers. If you see, the first flow cytometer was launched in 1970 by Beckman Coulter, followed by the first sorter in 1975. And over the years, we have come up with a lot of innovations. And in 2014, we launched our first Flex family cytoflex, which was based on which was based on WDM and APD technology, which is different from the conventional flow cytometers. We have almost around 7,000 installations globally, and with more than 500 installations in India, both in research and academia institutes. And this was followed by with the different Flex family modules that are available with us. The first module is Cytoflex with 3 laser and 13 colors, followed by Cytoflex S with 4 laser and 13 colors. And then in 2020, we launched Cytoflex LX. Oh, I feel this. So in 2020, we launched our flagship product, which is Cytoflex LX, which comes with 6 laser and 21 colors. So I would like to thank everyone for all the support that you have shown to us with more than 500 installations in India. And those labs who are more interested in studying applications like nanoparticles, exosomes, and EVs, we have a very dedicated system called Cytoflex Nano Flow Cytometer. Here, a lab can read as low as 40 nanometer size particles. And those labs who are interested in studying complex sortings, then we have Cytoflex LX with us. Now, continuing with the innovations with the Cytoflex family, we have detection modules that enables spectral capabilities, and that is called Cytoflex Mosaic. Sure, that's fine, not a problem. So this is a small, I think there's some problem with the pointer, but this is a small video that talks about how our existing Cytoflex can be integrated with a spectral module without any new instrument requirement, and thereby bringing conventional flow cytometer conversion into spectral flow cytometers. If we talk about inside, everything remains the same. All the optical fiber filters, WDM, and the bandpass filters are the same. The only change that is required is to change the optical fiber filters from the conventional flow cytometer to the spectral module here. And all the optical fibers are connected to the backside of the flow cytometer, to the spectral flow cytometer here. And all the bandpass filters remains the same with the same sensitivity what Cytoflex has shown over the years. And also we see that all these lasers which are available with the conventional have a dedicated side scatters. And there are different configurations which are available here for different modules, like Cytoflex S can be converted into Cytoflex Mosaic with 63 detection modules, and Cytoflex LX can be converted into Mosaic 88 detection modules here. This is how, in a lab, the spectral conventional microscope, conventional flow cytometer can be converted into a spectral microscope. You do not require any service engineers, but just with the help of your students, you can change the cables from the conventional to the from conventional to the spectral modules. And this cable is almost 1.8 meter. So this is connected with almost around with the 1.8 meter cables, which is present at the back of the instruments. And also, if we see, the length is sufficient enough to keep the module at the next to your existing Cytoflex S module or LX module, or it can be kept under the module also. And this cables has been tested for almost around 3,500 times, and there is no breakage, and it is continuing. So what Cytoflex Mosaic has to offer? The first one is sustainability, because there are different needs of the labs. And the next one is the flexibility, because it is very easy to convert from your existing flow cytometer to the spectral flow cytometer. Backup, when we talk about backup, it is all about you don't need any special panel designing, and all your existing panel designing can be converted into your spectral module here. And then easy to use with your Site Expert software, which is the platform for across all Cytoflex family. So if we see overall the Cytoflex Mosaic module here, the first advantage of it is the manual system can be converted into a spectral system very easily. The second is it comes with a Site Expert software, which is a platform across all the flow cytometers with Beckman-Coulter. Then it comes with 2 algorithms for spectral unmixing and results. Then it comes with 10 channels to eliminate autofluorescence interference effectively, and this is very important, especially when a lab is working with the complex molecules. And then all the lasers have dedicated side scatters, and that is how you can enhance your conventional flow cytometer to a spectral flow cytometer here. So that was my last slide. Thank you very much. So if you need any other info details, please visit to our stall. We are present at stall number 3 here. Thank you.
Thank you, Danaher Corporation, for a very well elaborative talk. Now we'll be taking a short. Sir, but the time allotted was 15 minutes. 5 minutes.
Good afternoon, everyone. My name is Akshay, application scientist from Molecular Devices. Molecular Devices is a leading life science technology company that provides high-performance analytical instruments, software, and reagents. Over the past few years, we have launched 30+ new products, 110+ patented innovations, 500K+ citations to support our technology. So Molecular Devices are mainly into three major business, that is bioresearch, drug discovery, and biopharma. In bioresearch, plate readers are the soul of our company. We have sold n number of single multimode plate readers across all over the world. We have our next generation ID5E, which comes with dual injectors for flash assays. And we have patch clamp technology for ion gated and voltage gated channels. And coming to drug discovery, high content imaging has made life simpler by giving us 3D images of cells, tissue, etc. And we have no competition when it comes to Flipper. Every company prefers Flipper to perform real-time kinetics. And in biopharma, we have QPix and Cell Select for cell line, protein, and antibody development. Next. Let us go back to the basics of cell culture and its challenges. Traditional 2D cell culture provides a simpler platform to study cell behavior, but lacks the 3D architecture and physiological complexity of real tissues. Organoid culture addresses this limitation by generating self-organized tissue-like structures and cellular heterogeneity and functional relevance. So culturing organoids have certain backlogs like handling, treating, and contamination, etc. Next. So to overcome this, Molecular Devices has developed three special platforms: Cell Express AI, SpectraMax ID5E, and Image HCS AI Advanced. Next. So this is the workflow where the organoids are cultured, maintained, and it is treated with a drug with the help of automated Cell Express AI, and the outcome can be read through either luminescence with the help of SpectraMax ID5E, or you can take the 3D images and analyze the volume or diameter of the organoids with the help of HCS AI. So this is a Cell Express AI. It's an automated cell culture unit. Next. So this is how it looks. And if you want to talk about the hardware, we have a liquid handler platform where it can culture the cells and it can feed and treat with the compound. And we have the media storage compartment where it can hold up to 2 liters of 2 bottles, and after the treatment or seeding or passaging, you can direct it to incubator where it can hold up to 154 plates. And if you want to image like cell organoids for like how it's growing or anything like that, you can, and there is an image system present there, so you can track everything with that. And the magnification ranges from 4x, 2x to 40x in that. Next. And this is how the liquid handler cell culture would looks like. So it has 8-span pipette where you can use it for like cell dilution or compound dilution, etc. And you can store up to 96 well plate and 46 well plate at a time. And there's a tip rack for where the tips are placed. And we have deck position where it comes with heating and cooling mechanism according to your assay requirements. And there's a tilting mechanism where you can aspirate or dispense the liquid without disrupting the cells. Next. So this is a microscope present in the system. It has confocal and autofocus system for better image. And it comes with the environment control, like you can run the live assays also. So this is how the software looks. Actually, biologists can build their building blocks just in few steps. Either it can be, you can like number one if you want to passage, or you can treat, or it can go for the imaging also. It is fully automated. There is no need of handling. There is no need of hands or anything like that. So if you once program it, it will do and it will tells you that it has been done. And if it is the confluence of 70%, it can tell you that okay, it has reached 70% or it has come to the end, so you can carry on further. Next. So you can track the cell growth and everything with the imaging system. Next. So this is the next generation ID5E. It's a multi-mode plate reader. Next. So it has 5 mode of detections. And we have monochromator and filter based for eye sensitive assays. And the filters are tagged with NFC, so there is no need of connecting the instrument or telling that the filter is injected or anything like that. So it has an error-free setup. And we have patented path check technology, which eliminates the difference between the spectrometer and the plate reader in the 9612 or 384 format. So we have smart dual injectors. It's like if you want to take reads with the kinetics or the kinetic, if the reaction starts immediately, you can capture that signals also using the smart injectors. And we have environmental control where you can run the kinetic assay with the temperature control or CO2 control or anything like that. And we have 2 adapters, and one adapter is for the micro volume where you can use the sample as minimum as from 2 microliter to 4 microliter. And we have the thing for like Western blotting also where you can be imaged or quantified. Next. So it has compatibility for 612 to 384 well plate, and it has a wide range of shaking options based on the user requirement. And we have something called auto PMT where the signal will not get saturated or anything like that. And super cool PMT, so if there is any heat in the instrument or anything like that, if you see the background noise altering your reads, it can be eliminated. Next. This is how the software looks. So you can specify what are the standards and what is the sample and unknown, and you can select either you want to go for absorbance, fluorescence or luminescence. And we have some predefined protocols also. You can just open the protocol and select your plate like 96 or 384 and you can run the plate. Next. So this comes with the CFR, FDA CFR on compliance. So it helps in filing audit trail or database, and it has individual user login, like when it comes to data integrity, it can keep the track who has checked the file or edited, anything like that, and it will have e-signatures with this. Next. 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 So these are the applications which can be done with the ID5E. Next. Next. Yeah, and this is the new instrument which was launched this year. It's a HCS AI imaging system. Next. So it has easy robot access where you can access objectives and plate, and bottom button is for external door. It can open and close. Next. So it comes in 3 modules. It comes in wide field, which has 5 LEDs without confocal. 2 minutes. Yeah. And confocal with LEDs and dual spinning disc confocal disc for the better resolution of the organoid or cells. And HCS AI Advanced, which comes with the lasers. It has 7 lasers and 8 channels. And it has a deep tissue spinning disc for better image of the tissue. It can go deep into the tissue, like it can tell the water, like the compounds are bind or it gets internalized or anything like that. And it can be easily modified and means upgradable on the site. So this is what I mentioned before. It 2 comes with a 5 color LED, and advanced comes with 7 color lasers. Next. Next. So this is how the hardware parts looks. It has 6 objective turrets which can be placed at a time, and dual confocal disc to eliminate any photo bleaching or photo toxication on the cells. And we have magnifier down, which can enhance the image into 1.5x. I'll tell you how it can enhance. And we have eye sensitive CMOS camera for better image. Next. So these are the objectives, which changes from 2x to 60x. It has long working distance and short working distance also. It comes with air and water. Next. So we have multiple spinning disc options. Lower the spinning pinhole size, higher the resolution. Next. Next. So this is a game changer in the HCS AI Advanced, because it is a magnification changer where you capture image under 60x. If you change the magnification into 1.5x, it can give the resolution up to 90x. I'll show you how. Next. So we generally provide 2x to 60x objectives. And next. So when you change the magnification into 1.5x, you can get up to 90x resolution. And we have autofocus system, which is hardware autofocus system and software autofocus system for the better image. Next. Next. So we can run the live cell imaging, which has temperature control and gas control with this. So this is how the acquisition software looks. Actually, you can set up the acquisition in the 5 simple steps. Select the plate, and you can configure which you want, object you want, and select the sites, select the channels, and it is ready to run. Second. Next. Next. Next slide. Next slide. So this is a AI-based Incata software where it directs the data into the Incata, where it can analyze and give the in detail about the cells. Next, you can. These are the CSAI application. Next. These are 2D applications. And next, last. So, next. Yeah. Thank you.
Thank you, Denher Corporation. Now we truly know why you are among the global leaders of diagnostics and life sciences. Now we'll be taking a short tea break. Speakers who have not submitted their presentation yet are requested to do so. We kindly request everyone to reconvene as we have an exciting lineup of sessions up ahead. Thank you everyone for having your presence in this seminar and conference. By 15 minutes. I request everyone to kindly be seated. We'll be starting with the sessions within 2 minutes. May I request everyone to kindly take their seats as we resume the proceeding to get ready for more exciting and insightful sessions ahead. As we now move forward to the next session, it gives me great pleasure to invite our distinguished chairperson to the dais. We are honored to have with us Professor R.S. Verma, Director of MNNIT Allahabad. Professor Verma is a visionary leader whose guidance and commitment has been instrumental in strengthening research, innovation, and academic excellence at our institute. I request Professor R.S. Verma to please take their seat in the dais. Joining him, we are delighted to welcome Professor Pawan Sharma, Adjunct Professor at IISER Bhopal. Professor Sharma is a distinguished name in the field of immunology and infection biology, widely recognized for his pioneering work on host-pathogen interaction and vaccine development. I request Professor Pawan Sharma to please take their seat on the dais. with both our esteemed chairperson now on the dais. I would like to request Professor R.S. Verma sir and Professor Sharma sir to take the charge of this session and invite the speakers for their respective talk.
Check, check.
Hello. Yeah. Okay. Namaskar. Namaskar to this session, plenary session. I am deviating from the program a little bit because I found that the logo of this conference is very interesting and probably full of information, but we have no idea what is being depicted there. So I will request Dr. Ambak Rai. Is he there? Oh, Dr. Ambak Rai will probably please explain to us what is there in this beautiful logo of the conference, as brief as possible.
Hello everyone. After the inaugural session, there were senior people, they asked me, what about this logo? So when the event was started, we were actually motivating ourselves. So we wanted to make something so that we feel motivated that we are connected to. So this particular logo, on the top you can see, the IMMUNOCON eye, that depicts the human proliferation, right? That starts from the zygote and the human proliferation. And since immunology for the human well-being, so I stands for the human well-being. And there you can find a bispecific antibody and a CAR T cell, and you can find the technology wheel, right? And the sustainability is represented by the 0 in the 2025, and there are 2 rivers which signifies the place where it's actually happening, right? So I think, sir, I have explained, and thank you everyone.
Okay, thank you very much. We have now some idea about this logo, beautiful logo. I'll request Dr. Professor Verma to please move forward. Thank you.
Good afternoon, everybody. I think welcome to the afternoon session. We have 2 speakers, Dr. Bali Pulendran and Rahul Purwar. About the speaker, Bali Pulendran is the Violeta L. Horton Professor Director of Institute for Immunity, Transplantation and Infection and Professor of Microbiology and Immunology at Stanford University. A distinguished leader in immunology, Dr. Pulindar's research focus on the uncovering how the immune system senses and responds to the vaccine, infection, and microbes. By combining system, biology, computational modeling, and clinical studies, his work has revealed key mechanisms that shape immune response and guide the development of more effective vaccine. Through his groundbreaking discoveries, Dr. Pulindan continued to advance global understanding of human industry and its role is human and health diseases. I welcome Dr. Bali.
Thank you very much for your kind introduction. I think I need help with the Mac. Thank you. All right. Let me see. Thank you. It's a great pleasure to be here. I'd like to thank the organizers for inviting me. Thank you for this place in Allahabad. I have to tell you that this is my second visit to this wonderful city. More than 40 years ago, my family and I, my parents and my sister and I, took this wonderful journey by train from Calcutta all the way to Delhi. And my father had booked us on the first class berth, but through some accident, we got traveling in the Janata class. And so it was a wonderful trip. I remember this very fondly. We stopped over in Allahabad for 2 days, in which we bathed in the Triveni Sangam, and all the sins that I had committed were washed away. And now I have to say that sins have re-emerged, just like SARS-CoV-2 rebounds with Paxlovid, so too my sins have rebounded. And so, unfortunately, on this trip, I will not have time to wash away the sins, but maybe next time. What I want to talk with you about is work we're doing in our lab, and the focus of my lab is to understand human immunology using vaccines as probes. Now, vaccines are incredibly powerful global health, public health tools. They save millions of lives, but they're also a wonderful way of perturbing the human immune system in a very synchronized way. So many years ago, my colleagues and I set up this approach that we call systems vaccinology, in which we use the tools of genomics and proteomics and metabolomics and so on to begin to understand the immune response to vaccination in humans. That, combined with computational approaches, has really given us a remarkable aperture into how the human immune response is stimulated by a vaccine, how it evolves, and what makes it durable, what are the mechanisms that result in protection against an infection, and so on. And so this field has really taken off quite well. And our initial work on this was with the yellow fever vaccine, which is a live attenuated vaccine, highly efficacious. It's been given to 600 million people worldwide, 99% effective. And so we use this as a model vaccine in humans to understand basic human biology. But what I really want to talk to you about today is the idea that the innate immune system in humans has a form of memory. Now, those of us who are immunologists, which I take as pretty much everyone here, understands the immunological memory is a fundamental aspect of immunology, of the immune system. So the cellular origin of immunological memory, we think, are memory T cells and memory B cells. But work in the last 10 years or so suggests that this evolutionarily ancient system of defense that we call the innate immune system also has a form of memory. And several years ago, we wanted to explore this idea that innate system has a memory by using vaccines as probes. And what we did was to use this H5N1 pandemic bird flu vaccine together with this adjuvant AS03. It's a squalene-based adjuvant. This is a vaccine developed by GlaxoSmithKline. And we immunized people with this vaccine, and then we collected blood samples from these people before they got the vaccine, and then 1 month or 2 months or 3 months after they got the vaccine. And we applied a technique called single-cell ATAC-seq. Single-cell ATAC-seq is a technique whereby you can look at the epigenetic changes that are undergoing in a cell type using at the single-cell level. And this is published here, but I'm going to summarize what we discovered. Basically, what we discovered was that in monocytes are an abundant cell type in the blood, and they constitute an essential cell type of the innate immune system. That in response to vaccination, monocytes were undergoing dramatic epigenetic changes that lasted for several weeks after vaccination. And so, as I say, this was published, but the question was, what kind of epigenetic changes are they undergoing? And there were two broad types of epigenetic changes. One was an antiviral program. In other words, the genes, the chromatin loci of genes that were involved in interferon regulation and antiviral immunity were much more accessible a few weeks after vaccination. But in the same cell, it's a single cell approach, other chromatin loci targeted by AP1 transcription factors. AP1 transcription factor, it's a family of transcription factors such as CFOS, CJUN, which are involved in the induction of pro-inflammatory genes like TNF, IL-6. So think of it like a pro-inflammatory program as opposed to an antiviral program. So what we discovered was that vaccination at once could open up an antiviral program and close the pro-inflammatory program. So the question was, does this epigenetic change have any functional consequence? So what we did was to isolate monocytes from these people before they got the vaccine at day 0, and then 21 days or 42 days after they got the vaccine, and then infect these monocytes in tissue culture with viruses like dengue or Zika. As you know, dengue and Zika are blood-borne viruses that have a myeloid tropism. In fact, monocytes are one of the main targets. And what we could show was that if you isolate cells, monocytes, from people at day 0 and at day 21 and at day 42, and then you infect them with either dengue or Zika, the ability of these viruses to replicate in vitro in monocytes was curtailed dramatically when you isolated these monocytes after they had gotten the vaccine. And this inhibition of virus replication correlated with these epigenetic changes that we're seeing. So that gave us some functional correlate of these epigenetic changes, that there is an antiviral imprint in these cells that lasts for a few weeks after vaccination, and this antiviral imprint is able to control the replication of unrelated viruses such as dengue and Zika. Now the question is this principle broadly applicable to many other vaccines beyond this H5N1-AS03 vaccine? So this is a study that we did over the last few years. We're writing up this for publication now. Basically, we've compared 6 different vaccines, including the original H5N1-AS03 vaccine, the H5N1 vaccine without the adjuvant, the seasonal influenza vaccine, the Pfizer-BioNTech COVID vaccine, live attenuated yellow fever vaccine, and the Shingrix vaccine, which is given with a very strong adjuvant, AS01. It's developed by GlaxoSmithKline against shingles. And I'm going to summarize the essence of what we saw. So when we looked at this broadly, we applied single-cell ATAC-seq to these data sets across 6 different vaccines, multiple time points, and we said, is there some organization to these epigenetic changes? How many different kinds of epigenetic programs can you see across 6 different vaccines? How are they regulated? Do they all go up together? Are they reciprocally regulated? So this is what this heat map summarizes the essence of our findings. So now this is single-cell ATAC-seq data. So you're looking at this epigenetic changes in blood monocytes, so one type of cell in the blood. And immediately you can see this red square here. So that is these transcription factors CFOS, CJUN. These are the AP1 transcription factors. It's red because the chromatin loci is wide open after vaccination. But you see, if you go up here, this blue box, these are the transcription factor loci of the IRFs, the interferon-regulated factors STAT1, STAT2. It's blue because it's closed. So immediately you see there's this reciprocal regulation. In other words, when the AP1 module is epigenetically open, the antiviral IRF module is closed. Now, if you come here, when the IRF STAT module is open, you can see the AP1 module is closed. And we also could define a couple of other modules, the CCAT enhancer binding protein loci and the NF-kappa B binding loci. And so this suggests that there's a certain organization in terms of how a cell type, like the monocytes, responds to vaccine and how its epigenetic program is organized. Some of these are coordinately regulated, others are reciprocally regulated. The other striking finding was if you looked at the classical monocytes, which we all know are CD14 positive, CD16 negative. It's a very seemingly homogeneous population, at least by flow cytometry. But if you apply single-cell ATAC-seq, you see at least 5 different populations which are shown here. C0 in blue is the ISG positive cluster, which is the antiviral program. And for example, you have the red one here, C3, which is the AP1 high program. Why this is so interesting is that if you look at different human beings individually, and each column here is one adult human being, okay? And you ask, what is the relative abundance of these epigenetically distinct clusters of monocytes each of us have in our blood. The results are really quite interesting. You see here in this individual on the far right, there's very little of the C0 or this ISG antiviral cluster, much more of the orange and much more of the red. But look at this individual on the far right. There's a much greater proportion of this antiviral program. This suggests that there's this incredible human variation in the epigenetic state of our innate immune cells from human to human. Now you might say, what is the functional consequence of this? Might this explain why some of us respond better to viruses in terms of a stronger innate program, stronger adaptive immunity program, and some of us do not? And this is something that we're currently doing. Now, just in case you're wondering, so is innate memory, like the one that I've described, is it all about epigenetics? I have to disabuse you of this now. I'm going to do a 180-degree turn and argue, no, it's not all about epigenetics. Because by nature, by looking at one cell type, like the monocyte, we're having a very myopic view of the immune system. The immune system, after all, is a system of interacting cells, T cells, B cells, dendritic cells, and so on. So the question is, how do these interacting cell types synergize or antagonize to create the property of memory or protection? So that's what I'd like to talk about in the remaining second half of this talk. And let me begin with this paper that we published a couple of years ago, in which we looked at this vaccine that you all know, BCG vaccine against TB. Bob Cedar at the NIH had done some remarkable experiments in monkeys where he gave this BCG vaccine intravenously and showed that after intravenous injection of the BCG vaccine, the monkeys were very protected against TB infection. And this is something that Jyoti is going to talk about as well, I think. And so the question was, what is the mechanism by which BCG given intravenously is able to launch such effective protection? And we were also interested in this idea. There's a lot of epidemiological evidence that people who get the BCG vaccine seem to be protected against a wide array of different infectious diseases. This is epidemiology, but we wanted to understand, is there an immunological basis to this? And so what we did, when I say we, it's Audrey Lee, a wonderful PhD student in the lab. She did experiments in mice where she gave BCG intravenously, and then at various time points, 7, 14, 21, 28, 42 days later, she infected these with SARS-CoV-2. This was done with Mehul Sutha at Emory. And this SARS-CoV-2 is the B.1.351 strain, the so-called South Africa beta variant, which has a high affinity for the mouse ACE2 receptor. And we measured the weight loss, and you can see starting at around 3 weeks or so, there's a substantial protection against weight loss. It persists for 28 days, 42 days, even at 3 months, although the effect is waning, but even at 3 months you see this significant effect. What's more impressive is when you look at the virus load in the lungs. So this is after infection, 7 days, 14 days, 21 days, 28 days. This is a log 10 scale. You can see at 21 days there's a 93-fold drop, 435-fold drop, 92-fold drop. So not a trivial drop in the virus load in the lung. Pretty significant drop, right? But that's in the lung. Now, if you look in the nose and the mice are infected with SARS-CoV-2 intranasally, you see there's no difference. In other words, BCG intravenously has resulted in a control of virus replication in the lower respiratory tract, but it doesn't prevent infection. So the question is how or why? And this is published, so I'm going to summarize what we understand about the mechanism in 3 slides, okay? So we did a lot of experiments to understand this, but let me just show you one type of experiment. We did spatial transcriptomics. This is a mouse lung of an unvaccinated, unimmunized, unchallenged mouse, a naive mouse. And you can see in green, it's a pancytokeratin marker. Those are the epithelial cells. In pink, you can see the few CD4 T cells. You don't see very many because this is an unvaccinated mouse. Now, look what happens a few weeks after BCG IV infection. There's this massive infiltration of CD4 T cells. Not surprisingly, we had shown by flow cytometry that these are Th1 cells that are making interferon gamma, CD4 T cells, BCG-specific CD4 T cells that are making interferon gamma, and you can gate on these CD4 T cells and ask, what genes do they express? And not surprisingly, number 1 gene, the top of the list is interferon gamma. Good, very good. Now, we can do a third group of mice where you immunize with BCG, allow the T cell response to develop, and then at some point, maybe 2 weeks or 3 weeks, inject an anti-CD4 antibody into these mice to deplete the CD4 T cells. Now you can ask, what impact has that depletion of CD4 T cells had on the genes that are expressed by these epithelial cells in the lung? Remember, these epithelial cells are the targets of SARS-CoV-2 infection, type 2 alveolar pneumocysts, right? So here we are, let's do the experiment. So this is a heat map. We're looking at a set of genes, which are the interferon-stimulated genes, antiviral genes. It's all blue in a naive, unvaccinated mice. On the far left, it's all red. This is 3 or 4 weeks after BCG, so it's blazing with this antiviral ISGs in these epithelial cells. Look what happens if you deplete the CD4 T cells. The interferon signature goes away. So we've done this, and we've shown that it's the interferon gamma that's being produced by CD4 T cells that is imprinting this antiviral program, and we've done other experiments to actually show this, but I'm not going to go through this in the interest of time. And you can take your favorite gene and you can do the number crunching, you can do the statistics, etc. It's really quite a striking effect. So just to summarize what we learned from this paper is that BCG intravenously is stimulating this very interesting effect on protection against heterologous infections such as SARS-CoV-2. This effect, we understood the mechanism of this was as follows. A few days, 1 day after BCG vaccination, you find this typical innate response that you see with any vaccine. You generally see this early on, but that goes away, and then what appears a few weeks later is this delayed phase of innate response that seems to last for a few weeks. Now, this delayed phase is mediated by the bystander effect of BCG-specific Th1 cells that make interferon gamma that feed back on the epithelial cells in the lung, but also other cells such as macrophages in the lung to stimulate an antiviral program. So we call this integrated organ immunity. Why? Because it's an integration of the innate and the adaptive immune system. So the adaptive, the Th1 cells, interferon gamma producing cells, feeds back on the innate system. So it's not one or the other, it's just an integration of these systems in the organ, okay? So Nature Reviews Immunology asked us to write this little perspective on this thing, and we wrote this and said, this might suggest a pathway to what we call a universal vaccine. We said that introduce the concept of integrated organ immunity to explain how the innate and adaptive immune systems can, and non-hematopoietic stem cells, the epithelial cells, can interact in tissues to generate enduring protective immunity against diverse pathogens in an antigen-agnostic manner. Considering immune responses through this framework could enable the design of a new class of vaccines termed universal vaccines that are not pathogen-specific. All right, it's easy to write a perspective, but much more difficult to do the experiment, to get the data to show that, right? So let's do the experiment. So what we've been working on over the last few years is to see whether the immunology we've learned from BCG could be applied towards developing a universal vaccine that could be given intranasally and that would have broadly protective effects against many different types of pathogens. And so we've been working with this company where we've designed these liposomes, which are for mucosal delivery intranasally. So BCG, we had shown in the previous paper, stimulates TLRs, toll-like receptors. All of its activity in terms of cross-protection was dependent on TLRs. In MyD88 knockout mice, this disappeared. So clearly you need a TLR ligand. So we've used 3MO52, which is a TLR7/8 ligand, and GLA, which is a TLR4 ligand. But it's not just TLRs, right? Because it's the feedback of the T cells, BCG-specific T cells that keep the innate system alive for a long time, the innate program alive. So we decided we would include some antigen, and we chose the world's best antigen, which is chicken, egg, ovalbumin, okay? So we did this experiment in mice where we give this in liposomes, day 0, day 7, day 14, day 21. You might say, why so many injections? Well, BCG is a live bacterium. It persists in macrophages for many, many weeks, perhaps months. It's constantly stimulating CD4 T cells to make interferon gamma. Whereas if you give an inactivated subunit vaccine, it disappears after a day. So we wanted to get the magnitude and the persistence of T cells that you might with BCG. Clearly, this is not translatable. You're not going to give this to a human at day zeros, but this is a proof of concept experiment to show, in theory, if you could do this, what would you get? So we infected this with SARS-CoV-2 at 21 days, 42 days, 3 months. You see very nice protection. At 3 months, the protection is waning. It's not significant, but there's a trend. If you look at the virus load, look, 693-fold drop in the live virus titers in the lung at 21 days. At 3 months, it's about 200-fold, okay? So it's a pretty significant effect. If you look at the lung pathology, you can see in the unvaccinated mice, there's a lot of damage and cellular infiltration. But in the vaccinated mice, the alveoli and the architecture is very well preserved. You can stain for the nucleoprotein against SARS-CoV-2. You can't see this in the vaccinated mice, and that's the quantification here. So what other kinds of pathogens could this protect against? We tried the seasonal influenza PR8 strain, 21 days, 42 days, 3 months, 7 months, and you can see a significant effect in weight loss, but also in survival, even as long as 7 months. What else? So with Ralph Barrick, we infected these with SARS-CoV-1 that protects against, it's a mouse-adapted SARS-CoV-1. Again, so it's very difficult to neutralize coronavirus, SHC014. It's protective against that. What other kinds of respiratory threats might you get? So what about bacteria? So we've shown SARS, SARS-CoV-2, SHC014, seasonal influenza. So we looked at Staphylococcus aureus or Streptococcus pneumoniae, 21 days, 42 days, 3 months later. And you can see, look, Staph aureus, this is the bacterial burden. At 21 days, it's a 14-fold drop. 3 months, 42 days, 14-fold drop. It's about a 6-fold drop at 3 months. Strep pneumoniae, 20-fold drop at 21 days, 66-fold drop at 3 months. Again, a pretty substantial effect. What other kinds of respiratory threats? So there's viruses, bacteria. What about allergens? So we looked at this with house dust mite allergen 3 months later, and you can see eosinophil numbers goes down, Th2 responses, IL-4 is down to baseline, IL-5 is down to baseline, IL-13 is down to baseline. The cellular infiltration in the lung architecture seems very well preserved in the unvaccinated, in the vaccinated mice, and the IgE expression goes down. So how on earth is this mucosal vaccine having such pathogen-transcending protective effects against viruses, bacteria? We've done it with fungi as well, and allergens. What's the mechanism? So we looked at the innate response. So this is FAC staining of innate cells, alveolar macrophages and dendritic cells in the lung. We look at CD86 expression, day 0, day 21, day 90 after the last vaccine. You can see in the alveolar macrophages, that is AM, there's sustained expression of CD86. You don't see this in the F480 macrophages. Maybe in the DCs you see it at day 21, but by day 90 it goes away, maybe a little bit in DCs, but really it's the alveolar macrophages. Now, what about T cells? So remember, we used ovalbumin. We've used other antigens, flu nucleoprotein and so on, but I'm showing you the ovalbumin. So we take the lung, stimulate them with ex vivo with ovapeptides, and then you do an intracellular FAC staining of CD8, CD4 versus interferon gamma. At day 0, there's really very little. But look, at day 21, really high frequency, 28%, 25, 35, 25%. Day 42, it's still high. And at day 90, it's still substantially high, 4%, 2.99%. And if you analyze this using class 1 tetramers or class 2 tetramers, it's really a persistent response, like what we had seen with BCG, okay? But are these effector T cells? Remember, BCG is a live bacterium inside macrophages, constantly providing antigens to T cells, so they're constantly making interferon gamma. Is this a live this is clearly not a live bacterium, it's an inactivated protein. So what kind of T cells are these? We analyzed them using CD45 intravenous staining to ask, are these tissue-resident T cells? We stained them with CD3 and CD109, as CD63 and CD69 and CD103. And in fact, the vast majority of these overspecific tetramer-positive T cells are tissue-resident T cells in the lung. They're not effector T cells, okay? So the question was, remember in BCG we had shown if you immunize and you eliminate the T cells, you lose protection. So what happens here? So if you immunize and before you challenge with COVID, what happens if you, well, let's do this experiment. If you don't have ovalbumin, so no T cell antigen, only the TLR ligands, right? Only innate, compared to if you have the T cell antigen. So if you do the two groups and if you look at the weight loss at 21, 42 days or 3 months, you see at 21 days in brown mice that received GLA 3MO52 plus over, very good protection. In blue are the mice that were unvaccinated but challenged, they lose weight or loss. And in pink are mice that didn't have the over. It's fine, they're protected. There's really not much of a difference between the brown line and the pink line here at 21 days. What about at 42 days? Aha, maybe there's a little bit worse. And what about at 3 months? Oh my, look, at 3 months you absolutely must have a T cell response. If you didn't have the T cell response, you lose this heterologous protection. We did this in a different way. We depleted T cells. In the interest of time, I'm not going through this, but we depleted the T cells from the beginning or towards the end, and we looked at this with every pathogen, with the SARS-CoV-2 infection, Staph aureus, the seasonal influenza. In each case, you absolutely must have the CD4 T cells and the CD8 T cells to actually feed back on the innate system. And so the question was, thank you, how exactly are the T cells working? So let's do the experiment here. Look, so if you immunize the mice and you look at CD86 upregulation in the alveolar macrophages, if you deplete the CD4 and CD8 T cells, look, it goes away. So the T cells are giving this signal that imprints the innate system. And I'm not going to go through this in the interest of time, but basically the signal induces an antiviral program. We've done this using spatial and single cell. It also enables these macrophages to take up antigens much more quickly. So you isolate the macrophages before you give the vaccine or 1 month after you give the vaccine, and then you ask how effective are they at uptaking labeled Staphylococcus aureus bacteria? Very effective compared to the baseline macrophages. At taking up apoptotic or necrotic cells that are labeled, very effective. Or taking up influenza-infected epithelial cells, very effective. So they have this remarkable property to take up viruses, bacteria, and dead cells. And they are absolutely essential. You can deplete these macrophages in vivo using clodronate liposomes, lose the protection against PR8 seasonal influenza or Staph aureus. I'm quickly going through this. The last point is, what signal are these T cells giving? Remember, these are not interferon gamma producing effector T cells. They are tissue resident T cells. We've neutralized gamma, TNF CD40 ligand. It's absolutely independent of these, unlike BCG, which is dependent on interferon gamma. What does matter is this molecule called rank ligand. Rank ligand is produced by these T cells in the first 2 or 3 weeks of their differentiation. And if you block rank ligand in vivo, you basically partially abrogate this protection against PR8 and other viruses. And the last couple of slides, you might say, okay, so this is a vaccine that you give intranasally. It's not a transmission blocking vaccine because there's still a ton of virus or ton of bacteria that gets in. 693-fold drop in the viral burden is a lot, but it doesn't go down to 0. 66-fold drop in strep pneumonia is a lot, but it doesn't go down to 0. So what does happen after these bacteria or viruses slip through this innate defense or this integrated organ immunity defense? What happens to the pathogen-specific immune response? So to look at this, we did this technique called Codex, where we can stain for 29 different colors by imaging. And this is looking at the lung 3 days after SARS-CoV-2 infection. You can see the odd B cells, cytotoxic T cell, helper T cell, in an unvaccinated mouse that has been infected with SARS-CoV-2. Now, what might this look like in a mouse that did get the vaccine and then a month later or 3 months later had the SARS-CoV-2 infection, and then 3 days, so 3 days is a very short time after infection. Well, what happens is you form these remarkable tertiary lymphoid structures. Looks like germinal centers, these ectopic germinal centers in the draining lymph nodes. And you can clearly see the yellow B cell zone here. You can see the green T helper zone and the cytotoxic T cells, and so on and so forth. And are these antigen specific? We can look at class 1 tetramers, and you can see after infection in the unvaccinated mouse in blue, the vaccinated mice in red, you can see as early as 3 days or 7 days, there's this rapid induction of the pathogen-specific T cell response and the antibody response, which I'm not showing here. So this is a double whammy, this mucosal vaccine. One, it reduces enormously the pathogen burden that comes in of the virus or bacteria. Two, for the pathogens that do slip through this initial barrier, it conditions the lung to make an incredibly rapid pathogen-specific adaptive immune response, okay? So let me summarize what I've told you here. Basically, intranasal immunization with this GLA3052 liposomes plus antigen provides protection against heterologous viruses, SARS-CoV-2, influenza, SARS, SHE014, bacteria, Staph aureus, strep pneumonia, and fungi. We've done fungi as well, and allergens for up to 7 months. Mostly 3 months, but with flu, it's up to 7 months. This protection is, for the short term, you can get by with the innate for 21 days. But for 3 months and 7 months, you must have the T cell response that's feeding back on the innate through rank ligand. I didn't show you the functional data. We can deplete the alveolar macrophages and lose protection. You can transfer the alveolar macrophages from an immunized mouse intratracheally into another mouse and restore protection. And there's this very rapid induction of ectopic germinal or tertiary lymphoid structures in the lung. I don't want to call them germinal centers, and tetramer-positive and antigen-specific antibody response. So we think that this approach represents a potentially new class of vaccines that we call universal vaccines. When we talk about universal vaccine, we talk about a universal influenza vaccine or a universal coronavirus vaccine. This is just a universal vaccine because hopefully it protects against many, many different classes of viruses, many different classes of bacteria, and even allergens. So it's a pathogen-transcending protection. Now we're doing non-human primate studies, and we're planning a study in humans where we can give this intranasally and deliberately infect these humans 1 month or 3 months later with flu to see what impact this might have in flu. So as I said, so I think we like to think of this as integrated organ immunity. It is a synergy of the innate, of the adaptive, and the non-hematopoietic epithelial cells to create this memory or protection. And I think it hasn't escaped my notice that in the Triveni Sangam, you have Ganga, you have Yamuna, and you have the mythical Saraswati all synergizing to wipe out your sins. So too in the immune system, you have the innate and the adaptive and the non-hematopoietic stem cells that synergize to wipe out a microbial sin. Thank you very much.
Thank you very much. Thank you, Dr. Pulendran. Wonderful, mesmerizing talk, I would say. And I'm particularly impressed by the extension of the durability of the immune response. Yeah. Relatively better one here. So may I invite some questions? Any questions, please? Yes, please, there. Piche.
Can I ask one? So I was just wondering how much this pathogen-specific T cells has to play its role for providing cross protection. Is it like the bystander T cell, which are actually providing protection by way of cytokine production?
Yeah, it's a good question. So as I've shown you that there's this remarkable induction of pathogen-specific T cells, I think they do play some role in protection. But we've looked at viral burden even before day 3, at day 1 or so, and even then there's a dramatic drop in virus load. So we think that whilst the pathogen-specific T cells contribute to this protection, we think that some of the early protection comes from the innate program that's been established in alveolar macrophages. So.
Yep. Great talk, as always. Girish Kirimanjeshwar from Penn State University. So this is reminiscent of Troy Randall's eyeball formation following influenza infection. Right?
Sorry, I can't hear you very well. Can you speak up a little? Yeah.
Can you hear? Can you hear me now?
Yeah, I can.
Okay. So first, I say congratulated on a great talk. This is a very reminiscent of eyeball formation from Troy Randall's group, like, mild infection with flu. So have you looked at any mild viral lower respiratory tract infection provides similar imprinting of alveolar macrophages and the following protection with a totally different?
Yeah, it's a good question. So we have done experiments where all these experiments so far I showed you were in naive mice, but we've done experiments where we pre-infect the mice with seasonal influenza, allow 3 months to pass, and then give this adjuvant to see whether even in a non-naive environment, this adjuvant has a similar effects, and it does. So we feel that the infection signal that's given, at least with flu, we don't know about other infections, but with flu, doesn't really quite set up the environment in the lung in the same way that this adjuvant is setting it up. Now, do other mucosal vaccines like flu mist, live attenuated flu vaccine that children get, is that doing the same thing? We don't know, but we want to test those ideas.
Yeah, I'm Kiran here. So here, please.
Yeah.
So what was interesting, nice talk. So it was like you say that it's a virus, it's an allergen, or it could be the particulate matter. But once it breaches the lung, the epithelial cells. So the thing is, when you give the liposomes, how does it actually activate the whole mechanism so that it is having either the alveolar macrophages that you are talking about has an immunomodulatory, or are you thinking about T regulatory cells as immunomodulatory? Because you are talking about even the memory T cells. So if I take it short, so how does the mechanism, basically the epigenetic regulation that you're talking at the germinal centers, or you're talking at the splenocytes? So where does that, the mechanism being modulated? Thank you.
Yeah, I think these liposomes are taken up by alveolar macrophages, and I think the TLR ligands in the alveolar macrophages has given them signal 1, the first signal. But that is enough to induce protection for 21 days, but not much longer. Signal 2 is coming from the elicitation of these tissue-resident T cells of any specificity, in this case of albumin. We've also done it with flu nucleoprotein and infected with COVID. And that signal 2 is important to provide an imprinting to sustain the capacity of alveolar macrophages to continue with this protection for 3 months, and in some cases 7 months.
Excuse me. That young lady there, please. Yeah. Sir, one of them. Iske baad puchna. Yes, please.
Thank you. Excellent talk, Dr. Pulendran. As a mucosal immunologist, I have one burning question. I'm Amrita from ICMR Nirbi. So your work, Bana Jabri's work, Yasmin Belkaid's work have shown that mucosal DCs function very differently. They have much more of a tolerogenic behavior, and in the context of diseases associated with epithelial damage, because the readout eventually is epithelial damage, whether it be allergies, viral, or bacterial infections. In that context, rank ligand secretion by CD4s or whatever, it can function directly on the epithelial cells for epithelial regeneration. What are your comments on that? Is it purely immunological driven, where the alveolar macrophages are behaving a certain way, or is this rank ligand production
great
activating
question.
rank signaling?
Yeah, thank you for that. That's a wonderful question. I don't know the full answer to your question, but I can provide with some mechanistic data insight we have. Yes, infections do cause epithelial cell damage, as you know, necroptosis, and other forms of cell death. The second point is that, as I quickly showed in the slides, the alveolar macrophages isolated from these immunized mice have a remarkable capacity to take up viruses, bacteria, necrotic cells, apoptotic cells, and virally infected cells. So they clear away the offending signal of dead cells, for example. And I think this is one mechanism by which they calm down the inflammation that might be caused by signals delivered by dead cells in the lung. And the question of rank ligand, in what way might that impact on this capacity? We haven't looked at this directly. What I think the rank ligand is doing is setting this system up so that these ectopic germinal centers, or these tertiary lymphoid structures that form so quickly, it's probably playing a role there. We've got some preliminary data on that, but that's still ongoing.
So I have
absolutely last question. Yes.
Yeah. So it was wonderful hearing from you, sir. And I'm Swatantra Kumar from King George's Medical University, Lucknow, and I work on viruses and viral immune responses. So I was just wondering, like a country in India, like most of the people, like we are living in an endemic country for TB, and most of the people have received the BCG vaccination. And you have shown the role of BCG as an adjuvant, I believe, and it is giving a protective immune response against most of the pathogens, including bacteria. So why, is it a T memory cell response which is giving the protection, or is it an initial IF and gamma response, which is, ideally we should not get an infection because we are already immune to BCG or TB as such?
Yeah. Thank you for the question. I think I got the question, although the acoustics are not so good, so I couldn't hear most of it. But I think you're asking about, in a country like India, where a lot of people have been exposed to BCG, and what might, in these humans, what might the mechanism of protection be? Is that yeah. I mean, I think it's a great question. Indeed, a lot of people, not just in India, but in many developing countries, have been exposed to either BCG or TB, subclinical TB infections. And we don't know, we don't understand the immunological mechanisms that are at play in the lung, in tissues, and how they impact on this kind of thing, on protective immunity. I think there's some data, but it's still largely a black box.
Okay, I have to cut down this session here because in interest of time. Thank you very much, Dr. Bali Pulendran. Thank you very much. And we move to the next speaker. It gives me great pleasure to introduce Professor Rahul Purwar, who is the pioneering star of cancer immunotherapy in India today. He is currently a professor at the Department of Biosciences and Bioengineering at IIT Bombay. He earned his PhD in molecular medicine from the Hanover Medical School, Germany. His PDF stint at the prestigious Harvard Medical School, Boston, Massachusetts, USA, and his association with IMMUNOCON, Inc., again in Boston as a scientist, underline the depth of his innovative science and the lofty heights of his courage as an entrepreneur. He joined IIT Bombay as assistant professor in the year 2014, a watershed year in the current history of resurgent Bharat, and pioneered the development of indigenous technology platform for chimeric antigenic receptor T-cell therapies, the CAR T-cell therapies. Based on research at IIT Bombay, Professor Purwar founded ImmunoAct in 2018, India's first cell and gene therapy company. Based on multiple successful clinical trials, ImmunoAct received market authorization of the country's first CAR T-cell therapy, and the Honorable President of India dedicated CAR T-cell therapy to the nation in April 2024. I understand this is the only first of the many planned milestones in democratizing access to advanced cell and gene therapies in India. More recently, he has been widely in news as the Honorable Prime Minister Shri Narendra Modi ji was presented the gift of 3 technologies at the Excellence Science, Technology and Innovative Conclave, ASTIC 2025.
I think that's good enough.
Yeah. And one of them was Dr. Rahul Purwar's humanized CAR T-cell technology. Well, immunotherapy comes in many garbs, and the one from which I have personally benefited is the intravesical BCG therapy for early stage bladder cancer. However, cancer immunotherapy as we know it today, this is totally, and pioneered by one of them is, pioneer is here standing before us, and that is completely changing the scene in the cancer therapy. So I welcome Dr. Rahul Purwar to present his talk. Thank you.
Thank you so much for such an elaborative introduction. And thanks a lot to the organizers, AMBAC and entire organizing committee to have me here and giving this opportunity to share some of our work. So I think previous talk was excellent, and a lot of science you learned about immunology, and here I will tell you about my journey in addition to little bit of the science
and little bit of immunology. Okay. And this journey basically is the result of last 11 to 12 years of developing some kind of hope to our patients who need state-of-the-art treatment. When I joined IIT Bombay as professor in bioscience and bioengineering department, I think one of the biggest challenge when I visited couple of hospitals are that many of our patients do not get treatment what they deserve. And majority of the times when you ask a doctor, what is the reason why we are not using these technologies, or these technologies are available, why aren't we using them? The answer is very simple, that it is not cost effective. It is the cost that determines the uses of a technology. So you can develop a wonderful technology on the world, but if it is not affordable, it doesn't reach to the masses, it doesn't reach to the people. And that instigated us to work on this particular journey, which I am going to share with you. We all have seen, I think, in the COVID time, taught us that India is the pharmacy of the world. We have provided our vaccine to the world. So I think in that sector, India has done excellent work. And actually, we are the pioneers in the chemistry-based small molecules. I think a lot of generic drugs, if you see the manufacturing, most of the world, I think, uses make in India small molecules-based drug, which are chemistry-based. In biologics, I think vaccine has given us a lot of, I think, goodwill. But there's a lot of work is needed in the biologics-based medicine. I think our neighbor, China and other countries who are doing a lot of good work in the biologics. When it comes to emerging new therapies, such as cell therapy, gene therapies, cell and gene therapies, these are intent to cure platform. The idea is not to just minimize the disease for some time, but rather cure a disease. At least intent is the cure. These platforms where industry is moving, all the developed world is moving towards this direction, the personalized medicine, precise medicine, and safe medicine. India did not have these kind of technologies until recently. We did not have it. And again, the major reason I can tell you is the cost. Why even we do not have these technology at any price. It's not like just only the rich people can afford it. In fact, doesn't exist. This particular technology which I'm going to talk about, cell and gene therapy, and this is specifically CAR T-cell therapy. She was Emily Whitehead. She was one of the first child who was treated at UPenn by Carl June and their team. And she has become basically now the ambassador of this particular technology platform, because when she was treated, she was a young girl, and now she is, I think, in UPenn doing undergrad there. Almost more than a decade cancer-free. And that gave the motivation that this particular technology platform has potential to cure a patient, which if you are a cancer physician, you will not like to hear the word cancer cure. You always say correct scientific word for cancer is remission. Patient goes under remission, but not called cure. But this is the first time when, so she goes every year to UPenn for this photo shoot, and every year you can see, and now this is the first time they are writing this cure. Basically, what I want to tell you with this slide is, take-home message is that with some technologies, patient can get hope, and there is a chance of curing the cancer. We are young in India, but we are also having, I think, many success stories now, which are not as old as decade old. Here is one of the professor from New Zealand, 62 years old, Richard. He traveled all the way from New Zealand to India to get the CAR T-cell therapy in Bangalore. And now he is cancer-free, 2 years, I think now. And he was giving, I think, some of the news channel. Not only one, I think another patient from Netherlands also flew to India, got the CAR T in Delhi, in the Fortis, and many more patients are coming now to our country and getting these kind of treatments in different hospitals with Make in India CAR T-cell therapy. This was one of our first child from Maharashtra, Nashik, and now 3.5 years passed. Fortunately, cancer has not come back. She is going back to the school, medical technician, and there are some more stories where they say he's a national cyclist champion who unfortunately got into this trouble, and there are many like dentists, some of our PhD student, or whether this gentleman from the elderly parent who is Bollywood aspirant and unfortunately got into leukemia, and now they are doing well. Ultimately, why we do science? We do all the science because we want to give hope to some people. Everybody will not get cure, everybody will not get benefit, but at least a section of the society, some people may get the access and may get some hope in these treatment. We are now, I think, having very high rate of cancer because as our economy is getting better and better, infectious disease coming down, and these non-communicable diseases are going up, and cancer in India is on the rise. And that's why it's a huge unmet need. And the major obstacle is cost, because all the treatment I have been talking about, whether cell and gene therapy, they're extremely expensive. And that's why what you see in the world map, India was nowhere. It's mainly the US and Canada, which is North America, and here you have in the neighbor China. Otherwise, you see neither in the African continent or the South America, Southeast Asia, almost nothing happening in this side of the world. So if you put a just simple line, then global North has these technologies, global South doesn't have any of it. And again, nothing to describe in this, that only the global South doesn't have because of the money. This is the real challenge. We are having a lot of patients in the world. In India, leukemia and lymphoma have similar frequency as other part of the world. So our patients numbers are more. So problem is here huge, but no solution available. The reason why I'm saying problem is huge because number of patients who die in our country, other part of the world where there are many other alternative technologies or therapies available. So patient has lot of different options, not just cell and gene therapy, lot of biologics are available. In our country, even many biologics are not available, and therefore, at death rate, people dying because of cancer is huge. I will give you one example. How these different, I am just putting cancer as an example because I've just taken this one system. Otherwise, you can think about autoimmune disorder, genetic disorder as well. So think about the axis problem. Brentuximab, this was a drug, again, monoclonal antibody, MAB, came to United States in 2011, approved by US FDA 2011, when Dhoni got the World Cup. By the time it reached to India, Rohit had the World Cup. It took 10 years, very simple 10 years, a decade, so that majority of you are from this part of the world, you understand that it takes almost a decade for a drug, even the monoclonal antibody MAB-based treatment, to reach to our country and helping our patient. Even when these treatments are great, again, excellent treatment, excellent drugs, they are saving human lives, but now you see here the cost of some of them. And these are the costs like $9,000, $10,000 for one cycle. And these are that, you know, that MABs you have to continuously take for longer time till the person is, cancer doesn't come back. Or they stop working, patient become refractory to these of the treatment. Then you have many other treatment where again the delivery is an issue. And therefore, there's a huge access gap, and we need CAR-T or similar kind of technologies which can cure cancer or other diseases much more than other part of the world. When you see 100,000 patients who are eligible for CAR T cell therapy in the world. For example, for one particular indication, if you take an example, and just for simplicity, I'm trying to explain it, 100,000 patients, for example, if they need it, less than 13% clinically eligible patients receive the CAR T in the reimbursement market. Reimbursement where like US and Europe, where these therapies are reimbursed by the insurance or the Medicare. But when it comes to us, less than 1% of our patients get it. So imagine less than 1% patient in our country are able to either afford it because we do not have this kind of reimbursement system. So what the world's offer now is just 15,000 CAR T, which are given to less than 13% of the available patient. In India and where the South America, you come to Middle East, or you come to our part of the world, these therapies are not available. And bottlenecks, again, cost is the major bottleneck, but then you need advanced and special infrastructure requirements, which I personally feel we have advanced infrastructure, clinical infrastructure for sure, to handle these patients without any doubt. These costs are not just for the drug cost. Just to give you a sense, the cheapest CAR T costs $500,000 US, which is 4 crore rupees for one patient. Then the manufacturing is again a challenge, and there are many aspects. So we had to fix this challenge for ourselves. And as any bigger problem, you can't solve alone. You need the entire team, and you need collaboration. So in this type of meeting, the best part what I like is networking, meet new people, and build some collaboration. So here, IIT Bombay, Tata Hospital, NCI, and IMMUNOCON. And here from Tata Hospital, Dr. Gaurav and Hasmukh, Nirali and Terry from the NCI. They were our knowledge partner. And Steve Heifel, also our knowledge partner for NCI for GMP work. And then IMMUNOCON, which I founded along with few others to take it to the masses. Now we also have many more people because team is growing. I am back to IIT Bombay, but at IMMUNOCON there are 250 people who are continuously working to help our patients. But in the team of advisors, then you need all of these people who actually pioneered the CAR T. I was mentioning Carl June, Satwa Nilapu, Dr. Ravindranath is Global Hospital. Some of you might have heard Global Hospital chain in India. He's the owner of it. Satwa is from MD Anderson. Gaurav, sir, as well as Hasmukh is from TMH, already mentioned. Dr. Avery Posi, again from UPenn. Nirali from NIH. Dr. Satwa Chava, who is the investor in the IMMUNOCON. We all came together to not just leave this technology at R&D level, but take it to the R&D to translation, and then translation to market, and now available across the country. So IMMUNOCON then gave the hope to some patients, and the innovation happening in the academia, like IIT Bombay, as well as Tata Hospital in Mumbai. And then the science with our vision that they want to become the leader in the cell and gene therapy translation research in India. And the mission remains to ensure the equitable access to all these innovative cell therapies to each and everyone who may get benefit from it. And that resulted for 11 years of journey. Ultimately, this first product, which is world first humanized CAR T cell therapy, this came out from the lab and then ultimately to after all the approvals. So this is now commercialized, available to all these patients who need it. And this we have done now many patients, even at commercial scale, almost 500 patients. Slide is slightly old. So we have done 500 patients with this drug. And now you can see the India map, which I showed in the first slide. There was nothing inside. And now you can see this is no more at least a boutique treatment. The CAR T is earlier what is called a boutique treatment, very complex technology, now available to all different hospitals, whether it's north, south, east, or west. Everywhere the CAR T is now available and given to the patients. it manufactured at Mumbai, and from Mumbai it is given to different hospitals for the delivery. We have ensured that not just tier 1 cities, in our country we have on the socio-economic basis, we have tier 1 city or tier 2 cities. Now you can see even tier 2 cities, patients are getting CAR-T-like treatment available to them. The idea is all hospitals, not just 1 or 2. So we have 90 different hospitals which are partnering with us, and entire like R&D work happens in the IIT Bombay, Tata and NCI, but not just in India. Now we are supplying CAR-T to even Mexico. Almost like it takes a lot of time to reach there, but now even Mexico, which is just sharing the boundary with the United States, our Make in India CAR-T is going there for the patients, lot of to Middle Eastern countries as well. So this product basically is now going to many places, I think. So from NextCAR19, yes, this was first one. We treated many, many patients, take it from the discovery, phase 1, phase 2, took to the market. Then another product just got phase 1 approved now after the discovery and everything. Now phase 2 will start very soon. Then the many other product in the pipeline. So with this, I will stop my talk, and if you have any questions, happy to take questions. And basically, idea is to give this innovation for everybody.
Wonderful talk, Dr. Nehru, wonderful talk.
Thank you.
Especially the affordability and accessibility. 500 patients already being treated.
Yes.
Yeah, any questions, please? Yes.
Yeah.
Please get up, yes.
Yes, many relapses. So it's not only the good stories. So as I said, not every patient will have the cure. There are many relapses, and like cancer will find its own way to come back. So there would 30 to 40% people will relapse. So the cure would be somewhere in the range of 40 to 50%.
Yeah, so we know there are a lot of success stories about the myelomas and blood cancers and all that. So any advancements for solid tumors?
Yeah, a very good question. There is a lot of work happening in the solid tumor, but you know, CAR-T, the unfortunate or the thing happened with the CAR-T is the benchmarking set very high. In general, if you see in the cancer medicine field, the response rate are 10 to 20% in general. Any drug you take, those are the type of response rate you get. CAR-T has set the benchmark of 70 to 80%. Even after relapses, 40 to 50%, right? Now in solid tumor, people try in glioblastoma and neuroblastoma. Their response is 20 to 25%. But given its very expensive treatment, now responses are only in the range of 20-25%. People are thinking CAR-T doesn't really work in the solid tumor. So, but there are many ways the people are doing science, and I'm sure that science will find its way. People will have better success in solid tumor. But at this moment, the solid tumor is only at the clinical trial stage, but no CAR-T got approved for market uses.
Okay. Any other question?
Yes. So, very nice talk and great achievements. So, because CAR-T cell was very expensive, what kind of the modification was done to reduce the cost?
Okay. So question is, CAR-T is very expensive. What are the modifications done to reduce the cost? See, there are two, cost and price of the CAR is two different things. Cost is how much it takes to make it. Price is at what price it is sold to the market. So what are the different drivers for pricing? One driver is manufacturing. But the second, the major one is IP. The development of that particular product is another driver. Then third is clinical development cost, which is huge, huge cost. So when a product is made, the R&D cost, In our case, for example, it is developed in academia. So the academic cost is low, and we don't need to pay too much royalties to anyone. So academia did the R&D part. Then translation into the clinical development, Tata supported a lot. Tata Hospital, Dr. Gaurav and Dr. Hasmukh Jain, they supported in the trial a lot. So the cost of running the trial, which goes in other developed part of the world, like $50 to $100 million, it was very small in our case. So clinical development cost was low. And the overhead, because of our infrastructure is not that expensive, other things, the manpower is not that expensive, overhead cost also is low. So IP, clinical development cost, overhead, and therefore the manufacturing cost is also low. So all these together make the cost low.
Yes, next.
Rahul, here, congratulations on bringing India on the map here. I just wanted to know from you the success rate of a patient that comes to you, you take their cells and all that, generation of CAR T cells and the success rate over there. And what are the factors that sort of influence that success rate of generating CAR T cells?
Yeah, that's also a very, very good question. In fact, we are right now facing that very big challenge in delivering the CAR T is whom to give the CAR T so that CAR T is more effective. And the reason it is very important in our country is that many patients, even the 26 lakhs rupees or 30 lakhs rupees even total treatment against 8 crore, we brought the cost to 30 lakhs. 8 crore to 30 lakhs, sometimes we feel very happy that we have done great work. But in our country, even 30 lakhs rupees is a lot of money. When a person gives 30 lakh rupees, they expect that results should be 100%, right? And that's exactly the question Dr. Mehra has, that what you should look into the patient characteristics or in the CAR T generation so this patient can get maximal effect. Unfortunately, this is more like clinical part of the question, and we do not know how to identify the best patient with 100% confidence. But there are certain hints, certain biomarkers which predict the prognosis of CAR T. One of them is tumor burden. So at what time you take the patient, if the tumor is already refractory to many drugs, you take those patients, then most likely CAR T will also not work. If you debulk the tumor and then give the CAR T success rate is very, very high. Then second issue what
These are all leukemias, of course.
Leukemia as well as lymphomas.
And the lymphoma.
Both.
Yeah.
So this is across the tumor burden, tumor bulk or debulking to get the CAR T success. But another major challenge what we have right now, that majority of the patient who are getting CAR T, they are all coming as a last stage patient.
Yeah.
And this is what we have to change, because once everything fails in the armory, that's the time doctor says, okay, now nothing is there with me, maybe we can try CAR T. Unless CAR T starts, doctors start planning clinical management at the right time for CAR T, because right now they have been using from last 20 years, 30 years, whatever their practices are, they have been using certain medicine. They have more confidence because they have been using it.
Yeah.
CAR T is a new kid which just came here in 2 years back. They are also learning and they are seeing how the science evolves. How to convince them that you start using CAR T at the right time, that only we can do once we have more data, more scientific
Just
data.
1 second more. You know the East Ocean Bio that I mentor, where we are trying to manufacture CAR NK cells and CAR gamma delta T cells. These are the kind of issues that we are addressing. What stage and who are the people who are going to benefit more? Because the cost factor is high.
Yeah, absolutely, absolutely.
Okay, any other question? One last question.
Hello.
Last question.
Sir, One quick question, Dr. Purva. So wonderful work, and I'm sure all the young scientists here would be highly inspired by this entrepreneurship. Now, when we talk about the heterologous CAR T cells which are available off the shelf, how effective they are in terms of not eliciting an immune response against those vis-a-vis the autologous CAR T cells?
Yeah, that's also a very good question, because autologous CAR T cell therapy comes with a challenge, because unlike the pharmaceutical drugs, what you generally take like paracetamol, etc., you can simply go and buy from the pharmacy. CAR T is autologous therapy. That means for every patient, we will take the blood, and for every patient, we have to manufacture the cells and give it to that particular patient. And this is so much logistic challenges. But good thing is it works. Now comes to the allogenic CAR T, which Dr. Luther is mentioning. Allogenic CAR T also works. There is a UCART program. There was a trial, and the company unfortunately shut down that program, not taking it further. But results were not that bad either. They were like 40% versus 70%. 70% in the autologous, 40% was in the allogenic CAR T, UCART program, UCART 19. In country like India, probably that's also a very effective strategy, because allogenic CAR T will be very, very cheap. How cheap, I do not know, but if you imagine if a patient is there and you have a choice of autologous as well as allogenic, and you tell a patient here you have 70% chance, and anyway that 70% chance, maybe 6 months if cancer come back, that reduced to 40%. But here you start with 40% and you never know, like at some point cancer come back and how many, etc. So that way it did not fly well in the developed nation. That's why I think allogenic program was shut down. And as you know, autologous really, really works, and now many success stories there for even a decade. But again, science is something great. That's what we scientists work. I am sure somebody will find the way out when UCART program will also start working.
Thank you very much.
Thank you
very much, Rahul. Wonderful talk and very good questions. We conclude the session here, and I thank both the speakers for giving very illuminating talks. Thank you so much.
Actually, we could finish on time because my presentation was the wrong one. But we did, I think.
You were well on time, much before that. Thank you very much.
Now I would like to request our respected chairperson, Professor R.S. Verma sir and Professor Pawan Sharma sir, to kindly present our esteemed speakers with the mementos. I request Dr. Rahul Purva sir to kindly come forward in the stage for the felicitation. May I also invite Professor Bali Pulendra sir to kindly come forward for the felicitation. These mementos are a gesture of our heartfelt appreciation for the invaluable insight that they have shared with us today. Now I would like to invite Professor Shivesh Sharma to kindly come forward and present the token of appreciation to our respected chairperson. Thank you, sir. As we move forward, we now come to a very special segment of today's program, the felicitation of senior members of Indian Immunology Society. This session will be conducted over the next 15 minutes, and while we truly recognize that a lifetime of contribution cannot be captured within any time frame, but we humbly request you to kindly cooperate and adhere to the allotted time. May I now invite our distinguished chairperson for this segment, Professor Amit Awasthi, sir, the President of Indian Immunology Society, and Dr. Kaustav Nayak, Secretary of Indian Immunology Society. I request our chairpersons to kindly come forward to the stage and lead the felicitation ceremony of the senior IIS members.
Thank you. So this is a special session, and this is to honor the contribution of our senior immunologist who contributed tremendously to the growth of Indian Immunology Society. And this is our duty to actually felicitate them, honor them, and listen to them about their contribution. So I will hand over to Secretary Kaustav Nayak to start this ceremony.
And thank you, Amit. So this is in continuation of our duty that we have been doing from quite a few years now. And last time we had honored 4 of our senior immunologists, and this time we have 3. The first senior immunologist we want to honor today is Dr. V.D. Ramanathan. To introduce Dr. V.D. Ramanathan, may I invite Dr. Aravindan, a colleague of mine and who has worked with Dr. Ramanathan for a very long time. So, Dr. Aravindan. So, please say a few words about Dr. Ramanathan before we can do the honors.
First and foremost, I would like to thank the Indian Immunology Society as well as the organizing committee for giving me this opportunity. Personally, I feel extremely happy to introduce Professor Ramanathan for 2 reasons. One is personally I know him for a very, very long time, and then he has seen me as a student and now he sees me as a principal investigator. I and Kaustav, both of us are from NIT, and when we were doing our PhD, he was an active researcher at pathology department in TRC. Just to highlight few important things about Professor Ramanathan, he is MD PhD, and he has actually done MD in pathology and PhD in immunology, a very, very rare combination in those days, and from the London College. And after that, he started working on complement. So when we were students, we always used to refer to Ramanathan sir as complement man for 2 reasons. One is, whatever questions you have pertaining to complement, he would be the best person to answer that one. And all his answers have always been very precise, practical, and enabling us to do the experiment. Second thing is, he always compliments the PhD scholars there with his goodwill and knowledge. One valuable thing as students, me and Kaustav have learned from him is, knowledge is universal and never restrict knowledge only to your students. So we always seen him only as a mentor, even though we are all Dr. P.R. Narayanan student. Any doubt pertaining to immunology, whether it is animal experiments or human models, we would just run to Professor Ramanathan, and he was extremely accessible to us. And even if he doesn't know the answer, he will say I'll think about it and get back to you. And many times, even if he's not able to give the right answer, he will give a clue which would enable us to go read back and solve the problem. So that is a very, very interesting, important attribute which I see in him. And coming to the scientific contribution, he has more than 50 papers, and he has been president for many prestigious immunology-related societies. But the most important thing is, during the fag end of his career, he actually took active initiative in IRV trials, which was conducted at TRC. So this trial was conducted to evaluate the HIV vaccine. At that time, it was a very big use, and he devoted all his energy, enthusiasm, everything, to make sure that the vaccine trial is conducted and completed perfectly, which he achieved. The only thing which, as students in those days, we felt was, this should have been given to him at a much earlier stage, where he had more energy and was more active. With these few words, I would like to request Dr. Kaustav to invite him.
Dr. Ramanathan, may I invite you?
I would also like to invite Dr. Usha Ramanathan, Dr. Ramanathan's wife, on the stage. We are sure that all of us, Dr. Usha Ramanathan has equally supported sir in all his work while he was in the lab. The next senior immunologist we are going to honor this evening is Dr. Utpal Sengupta. To introduce Dr. Utpal Sengupta, may I invite Dr. Itu Singh, who has been Dr. Sengupta's student and now a colleague.
Good evening, everyone. I am very honored to talk about Sengupta sir on this forum and about his work, his honors. And I'm the student of Sengupta sir, and I worked I know Sengupta sir from 2000 till now, and I'm working still with him. He is a renowned immunologist and leprosy researcher, and he worked initially in the PGIMR, Chandigarh, as the pathologist, and he did his PhD in the pathology. Later, he joined ICMR as the senior research officer to develop the Department of Immunology in Jalma. And then he joined Jalma as the director, and he worked as the director in Jalma till 2001. And I used to know him from the 2000. I did my MSc from there, and afterwards I joined as the PhD scholar under him. And I completed my PhD from there, and then I joined Stanley Brownie Lab, and he was working with the Stanley Brownie Lab, the Leprosy Mission Trust India, from 2011. And his major research area are like autoimmune phenomena in leprosy, monoclonal antibody, use of monoclonal antibody as the diagnostic markers in leprosy, early diagnostic markers for leprosy reactions, and development of early diagnostic tool for the diagnosis of leprosy. So still we are working on the this validation of early diagnostic tool for the diagnosis of leprosy in the field. So he is guiding us with us, and he is very much associated, very helpful for us, and he always used to tell us what to do and what not to do. So I'm very fortunate to work with him. And his key contribution include like Dharmendra lepromin antigen and developing monoclonal antibody based serodiagnostic assay. And his publications are many, like over 239. And he has his honors and awards include Erwin Stendhal Memorial Oration, Jalma Trust Fund Gold Medal, and Professor R.R.K. Shukla Award, Fellowship of NASI and NAMS, and IAL Lifetime Achievement Award. So I welcome Sengupta sir on this stage.
third senior immunologist to be honored this evening is Dr. Sunil Arora. Let me ask Dr. Amit, who has been a long-time associate of Dr. Arora, to introduce Dr. Arora.
Thank you, Kaustav, for giving me this honor to introduce the one and only one, Professor Sunil K. Arora. My association with Professor Sunil K. Arora started in 2016 in one of the IMMUNOCON meeting in Visakhapatnam, where I see a gentleman who was really fearlessly really saying about how to improve the Indian Immunology Society and what are the steps we need to take. And that's for the time I came to know, oh, he's the one, he's the Sunil Arora. And then in 2018, when the election was about to happen, I called him in one of the evening and I say, sir, I met you in 2016 and you were saying about few things. Let's change it, and if you want to change it, we have to be in the helm of the affairs. Why don't you become president and I come with you as a secretary? And he was very reluctant to accept this offer. And then he said, we'll see, and I think maybe it was a post-dinner time, so he may be tired. He was really not really responding that positively. But then he called me and said, okay, let's do it. And from that time onward, we were like associated together. And his contribution to the Indian Immunology Society is enormous. He has been a member of the society quite some time, maybe 40 years. And then he was a joint secretary of the society, vice president of the society. And 2018, he became the president of Indian Immunology Society for 4 years. And then he was a vice president of FIMSA, which is a kind of international body. And then he became president of FIMSA also. And he has contributed in terms of organizing various meetings and the conference, World Immunology Day, FIMSA Congress, not FIMSA Congress, the FIMSA course twice. He organized 2 IMMUNOCON meetings. One is in 2006 and then other one is 49th meeting in the Chandigarh. He has been contributing a lot in the research, which is reflected by his publication. 180 publication he published and 25 PhD student from his felicitate him for his contribution. I request Professor Sunil Arora to come on the stage and be part of this journey what we are doing together. Thank you.
Let me invite all the dignitaries on the stage, Dr. Aravindan, Dr. Ramanathan, for a group picture. Dr. Sengupta, Usha Madam, Itu. Dr. Itu is not around.
Dr. Amit, would you give me a few seconds to say something on this? Namaskar, everyone. Good evening. And I really appreciate the executive of Indian Immunology Society to choose me as one of the senior, because मुझे लगा, अमित को लगा कि जल्दी से निपटा देते हैं इसको, दे दो आके बाद में पता नहीं क्या होगा इसका. But what I want to say is, I have been associated with this Indian Immunology Society when I was very young, you know, a young research scholar. 1984, I became the member of this society. And I have seen stalwarts like Professor G.P. Talwar, at that time was a very senior professor and head of biochemistry in AIIMS. And he was conducting all those workshops, and we used to go there and learn so many things. And then there were so many others, and Dr. Sen Gupta and all. It was an honor to be chosen among these stalwarts, you know. But I developed a sort of liking for this society, and I've always tried to attend the meetings and tried to see how we can bring some reforms in the functioning of the society. We have seen it growing bit by bit, and when International Congress was conducted by Professor Talwar and Professor N.K. Mehra, it was kind of an honor which came back, and I always thought, why can't we bring it back? And when I became president of FIMSA, the first thing I told Amit, we have to bring some big meeting to India back, and now we are bringing FIMSA Congress next year. Amit Awasthi will organize that in 2027. It was only during my tenure that we proposed it, and we are bringing back FIMSA Congress at least. And I'm sure Amit Awasthi and team and many other young people who are now coming up, they will bring International Congress also. Once again, when we will have Professor Talwar among us as a centenarian, and he will basically inaugurate that Congress. So thank you very much. It's a lovely feeling to be honored on the stage of Indian Immunology Society. And I thank everyone in the society for all the cooperation and lovely meetings, you know. I wish the society all the best. Thank you very much. God bless.
Thank you. I'm overwhelmed with all this honoring me. But anyhow, I'm still with the subject, working on. I'm attached to a leprosy mission, and I started leprosy work long ago in 1976. 74, as a postdoc in London, and 76, I started developing immunology lab in Jalma. Professor Dr. Dr. C. Gopalan just told me to develop the immunology lab. So we developed it from scratch, and there we worked for throughout our, up to the retirement I worked there. A lot of PhD students passed through, but anyhow, ultimately leprosy I could not leave, and leprosy mission called me, and I'm working still with them, developing projects on leprosy. And from the very beginning, we had a desire or attempt we made to develop a test for early diagnosis of leprosy, which was very difficult because it's a cross-reactive organism. It's very difficult to make an antibody-based assay. So we went to the molecular level, and now we have got and developed a test for early detection of leprosy, even in subclinical stage. And it is with a company called TrueNet, where TB system is there, and COVID is also there. Only cartridge you will have to change, and you can do the test on the spot and it will be, if government takes it, it will be planned to be launched. Anyhow, thank you very much for recognizing. I am always associated, I was always associated from the conception, the day it was formed in the All India Institute of Medical Sciences, where Professor Talwar formed the Immunology Society. From that time till now, I am seeing it growing, but it should grow at a level as a subject. Biotechnology has grown, but immunology as a subject, it has still not grown. And I don't think it has been percolated to the common population. So we should try about that, and Dr. Santosh is taking steps for teaching immunology to our community-based type to understand it, how population can be helped by the knowledge. Thank you very much. Thank you very much for giving this honor. Thank you. Thank you all.
Mr. President, Secretary, friends, and colleagues, I have only two emotions. One, I am overwhelmed and humbled. I'm not sure that I have done enough to get this honor. At the same time, I was overwhelmed for another reason. It's because the honor was given along with Dr. Sengupta, whom I revere and whom I consider as one of the topmost immunologists and original scientists that I have ever encountered, not only in India, but elsewhere too. Just as an example of that, in 1980 when I joined JALMA, one of the first things I saw him was, I was coming into the lab, he was going with a syringe. I said, where are you going? Oh, this is emlepreg from the mouse. I'm going to inject it into the banana tree and then cut it at various times to see the viability at various times. And he has done original work on lepramine. I think that kind of out-of-the-box thinking is needed for this society because, and also, it's not just the immune system alone. We should not put blinkers. For example, I'm sure Dr. Pulendran also will concur. There are many other things which happen. In fact, one of the papers by Dr. Aravindan also shows the immune endocrine axis. Please broaden your vision. That's my request. And think out of the box. That's the first reason why I was overwhelmed. I was also happy, and I would like to make a public acknowledgement that Dr. Usha, my wife, also was called. She is a qualified psychiatrist. And so in the evenings on many days, as you know, in experimental science, most of the days the experiments won't work. And I'll come home with a very sad face. She will put on her best clinical manners as a psychiatrist and say, now, what is it? Come. And then give me a cup of coffee, and then I'll go fresh for the next day's experiment. So that way, I'm really very happy and very grateful to the Immunology Society. And I wish all the best. And I'm sure within the next 10 to 15 years, or hopefully even less time than that, there should be many Nobel laureates coming from India doing work on immunology. Thank you very much all.
Thank you. It's over to the organizers.
Thank you, Professor Avasthi and Dr. Nayak, for leading such a wonderful felicitation and your inspiring leadership within this IS community. Let me take a moment to reflect on the legacy of MNNIT Allahabad. Established in 1961, the institute has grown from a pioneering regional engineering college into a National Institute of Technology and an institution of national importance, with the Department of Biotechnology taking shape in 2006 and flourishing ever since. The institute has expanded its footprint into the life science. And today, as we stand at the intersection of science and innovation, it is a moment of pride of MNNIT Allahabad to host IMMUNOCON 2025, celebrating excellence in immunology and advancing the frontiers of research. Now, we lead on to one of the most engaging segment of IMMUNOCON 2025, the industry-academia interaction. To lead and moderate this insightful discussion, I have the honor of inviting Professor N.K. Mehra, former professor and dean of research at AIIMS New Delhi and national chair scientist at ICMR. Please bring a huge round of applause as he comes to the stage. Joining him for this important interaction, we have an esteemed panel representing both academia and industry. First, I would like to call upon Professor Suman Chakrabarti, Director of IIT Kharagpur. He is an eminent researcher and a recipient of the prestigious NASI Reliance Industry Platinum Jubilee Award for application-oriented research. With him, Professor Rahul Purwar, IIT Bombay, and the founder of ImmunoAct. He is a leader innovator in cell and gene therapy, known for his pioneering work in India's first indigenous CAR T cell therapy. Now I invite Mr. Chaitanya Gundu, country leader at Bachman Coulter Life Sciences and commercial strategic director at India LSIG. With him is Dr. Shirisha Potala, Associate Director of Strategic Planning and Operations at Amgen. She is a seasoned leader driving innovation and excellence in global pharmaceutical strategy. May I request to all our panelists to kindly come forward and take the seats on the dais. We look forward to a thought-provoking and collaborative discussions ahead.
Thank you very much. May I ask the people who are at the back to come in front, please? At the back, come in front. Thank you. I think this is going to be a very interesting talk because I think India is now moving to a stage where we want to expose our youngsters to industry as well. During our time, it was a taboo. If I worked all my life at the All India Institute of Medical Sciences, New Delhi, and if I was invited by any private organization to be a member of their scientific advisory board, it was denied that you cannot actually pass on your knowledge that you gain from AIIMS labs to any private enterprise. I think times have changed now. And the times have changed, but particularly because IIT showed the way. IITs had this kind of a interaction, and of course in our own subject we have seen all over the world, in the US labs or in the European labs, all the professors were actually encouraged to establish companies or do something else. And you saw every time a new journal comes up over there, because for somebody who was to go up the ladder, you should have your own journal, you should have started a new scientific journal, or you should have started a company. The reverse was the case in India. And I'm so glad that the Indian Immunology Society has now decided that every year, and that was started, I think, from our Golden Jubilee time when which Uma and Sujata, they both are there. They organized the Congress in AIIMS 2 years ago, that we should have a session devoted primarily to academia-industry collaboration or interaction. Academia is involved in generation of knowledge, and we are training our students to only publish 2 or 3 articles, and that's about all. we actually don't expose them to industry. Isn't it the case in myself? I got retired from AIMS in 2014, and of course I had to join the industry now. Or I could join any academic institution, but I decided to also have a taste in the industry. And the kind of things that I saw over there, I had never been exposed to. It was something new for me. My feeling is that now time has come during the PhD time itself, we should send our students for 2 to 3 months of internship to any industry there during their studentship time. Because they must know, we are training them to take a degree and then you look for a job. Job nahi hai, itni jobs nahi hai. They keep loitering around and all that. Find a postdoc fellow, somebody from America comes or Europe comes and they move after them and all that. Oh, I want to do postdoc and all. Because there were no jobs. Now we want to train them to create their own jobs. You know that in 2014, and I as the Vice President in Indian National Science Academy had been talking about it also through our G20 forums. In 2014, India had only 100 startups. Today we have 150,000 startups. And more than half of these are in the biomedical field. And even if half of them succeed, of the type that Rahul talked about, I think India is set at a pace that in the next 5 years or so, industry will boom in India. So I don't want to take too much of the time because they have only allotted 45 minutes. I will ask each of you to speak about your experience. There are two guys who are from the academia side, both from IITs, and there are two here who are from the industry. So I'll start with you, Suman. Talk about your experience in the IIT Kharagpur and how the IITs actually came forward to encourage academia-industry collaboration.
Yeah, this is a very important question, and not just from the viewpoint of the focus of this particular conference, but overall, be it IITs, be it any other academic institute, the classical approach of research in India has always been centered around academic pointers, and academic pointers are typically those pointers which will make one progress in the academic ladder. For a student, it is publications. For faculty, it is again publication, citation, number of PhD guidance, so many other parameters. But the translational aspect of the work, I think the major challenge with the system was that people have always been doing translational work in the Indian academia, in IITs and IISc and many other institutes. The problem is not that. The problem is that it has not been organically accepted as a means of career progress. That means a student who does not per se publish any paper, but spins off a startup which gets into a good stage within the 3 years or 4 years of the PhD, are we in a position to give the person a PhD on the basis of evaluating that? I mean rigorous evaluation, I am not saying just superficial. Or someone who has not published lot of papers but has got significant technology transfer which have got commercialized. Are we in a position to think that as a acceptable promotion channel for a faculty when the faculty member may be short of other things in the criteria? These kinds of things where the career progress of anyone, if they are linked with industry academia connection in whatever way, it could be through consulting work, it could be through R&D and innovations, not necessarily getting the final product into the market, and also lab to the market. So if these things are brought into the ecosystem, where the ecosystem is not just having these as elements, which currently it is much better than what it was 10 years back. We have all these things in the ecosystem, but I think what is still there missing is that how these are considered to be pointers of progress for the students, faculty, etc. And that what you are mentioning, that possibly that is for the reason that a PhD student while graduating is looking for someone who will give a postdoc position abroad, instead of using the opportunity of the know-how that has been created in the PhD to translate that into commercialization cycle. Well, everybody may not be talented in that, but I think there are quite a few who are talented, but they are not really having that systemic encouragement towards that. I think that's a major issue.
I'll come back to you for the second round. I should go now to the industry. I didn't introduce you because everybody knows you, but because you gave a talk. I must move to the industry person. Ladies first. This is Dr. Sirisha Patala. She is the Associate Director, Strategic Planning and Operations at Amgen India, leading externalization strategy for biological development.
It's nice to be here. I think to begin with, I'm an
Tell us
alumni.
first, how did you move from academia to industry and why?
So I'm an alumni of IIT. So I did my PhD from IIT Madras. Again, very similar to what we know at that time. We had just two choices. Get into academics. For that, you have to do a postdoc, get into teaching. Second, get into industry. The depth of industry or the diversity in the industry is something which I didn't know. But then, even in industry, we say something, it's okay to fail, but fail fast. Don't delay in failing. So I took a similar approach, knowingly or unknowingly. I went for a postdoc abroad, a very traditional approach. Once you finish your PhD, you apply for a postdoc. I did that. But 6 months into the postdoc, I realized I don't want to get into academics. So I came back to India. I was very clear on what type of companies I want to join. I shortlisted my own companies, and I started applying only to these organizations. 15 years I have been in the industry, and today, again, to add to what Sir has been telling, industry and academia are two different worlds at some point. But if we look for the last couple of years, things have been changing. So last month, actually, IIT Hyderabad has invited 15 industries to relook and design their curriculum to make the workforce industry ready. So that's a big step. That was something which didn't happen in the past. And then I think just last week, a week ago, I was in IIT Bombay talking to research scholars, showing them actually within industry what are the different verticals that are there. Because as he said, I'm leading a strategy part. So what does strategy do? How does my science background help in strategy? How did I get into the industry? How did I navigate? Because sometimes what happens is when you do a PhD in science, my PhD was in cancer biology. I was working on immunotherapy. When I entered as a scientist. Eventually, I found an interest and inclination to industry. Within industry to business aspect of it. Basic questions like why the industry is deciding on a certain target. I navigated. And then I realized you actually don't need an MBA to be in business. It's just your inclination, right mindset. So a couple of years in science, then I became a team lead. I made a lateral move to business. Then I moved into strategy. So there are a lot of opportunities within industry. And there's a lot of industry academic collaborations that are going on, starting with internships, starting with relooking at curriculum. Things are changing. Things are evolving. Yeah.
Thank you very much. I know there are challenges and demands both in industry as well as in academia. I want to give you one example. I was the dean of the institute in 2014, and we used to get a lot of pressures from the Holland universities there. There are 5 of them. They were very keen to develop collaboration between them and AIMS in Delhi, actually. By the way, now there are many AIMS. Earlier, we used to call, AIMS would mean only AIMS Delhi. Now I have to say AIMS Delhi. And I took a delegation of 5 of my very senior faculty members really the top class. And we visited all of those 5 universities there. We were surprised in each of the university, there was a separate building, a new building. It was like an incubator there. It was run by the industry. Industry was right inside the academic university there. And they said, anything that you do in the labs, that you find something good, you don't know what to do. And I really regretted if that kind of situation was there in AIIMS for us, many years, we would have several of those. Rahul, I want to come back to you because you have tasted both. You have tasted the academia, you have tasted the industry as well, and you have established the company as well now. So tell us your experience and make sure that there are a lot of youngsters over here who would like to emulate you.
Yeah, thank you so much. Yeah, my experience has been, I think, great. It has been a good journey. So I started at IIT Bombay, fortunately, which allows or which gives you the platform for the incubator. IIT Bombay incubator is already, I think, 25 years old. So we have that culture from long time. It's maybe not there in many government institute, but IITs has this culture of translating your lab research, and faculty has given the freedom to start the company as well. So faculty can own the company and can incubate in the incubator of IIT Bombay. And but of course, they can go on the lien and can also run the company for some time or whatever the time is needed. So we have that academic freedom to start a company, to run the company as executives, as well as now, but once you come on back after your lien, then you cannot have executive post. Then you can still remain the founder, etc. So we have the system, and IIT has this culture. I am not the only example, there are many. So there are many professors who have started the company and successfully those companies are running now independently. So I think India has the ecosystem. So we have the ecosystem. It is possible that many other institutes don't have that IIT Bombay or ISC-like system, but system exists. If other institutes want to take it, I think they should take it, in my opinion.
Yes.
Right? And yes, there are challenges, especially for students. Yes, having a startup in our country is a challenge, of course. There are many levels of challenges you will feel in industry as well. For me, if you ask, even my own students do not want to join the industry. If you ask them in the beginning, I used to ask them, would you like to join? And there would be no, I want to become the professor. So I think in our culture, the way we have been raised, the way we have been, I think, growing or learning in academia, I think culture is that if you want to become in science and engineering, I want to become the professor. This is, I think, last year for the first time my own student said, I want to join ImmunoAct.
So Chaitanya, is that the easy way that the youngster is trying to look into that, oh, okay, I have done this and now I can run up to the
I don't think it's an easy way. I think youngsters are very hardworking, but the way they have been raised, the way they have been listening classes, the way they see their idols, who they look at. I remember seeing you, for example, being the one of in the AIIMS, and we would say, I want to have a facility like Professor Mehra, right? So then you want to become the professor at AIIMS. That's how your mindset is. I think there are great examples. If you see Indian biotech, they have a pharma, like chemistry-based I was mentioning. I think many people who are studying NIPERS, they go in the pharmaceutical industry.
Yeah.
We need more and more R&D-driven startup, R&D-based biotech company, and I'm sure our youth will take those kind of careers forward as well.
No, here what we are talking about is academia-industry interaction.
Yeah.
So that the academia, they should also develop a project in association with industry. And not that the two has to work in the silos there. I want to come back to you, Dr. Gundu, Mr. Chaitanya Gundu. He is the country leader of Beckman Coulter Life Sciences, a company that has been there in India and around the world for many, many years. And, we have always talked about flow cytometers from you. So tell us, how could industry be a partner in knowledge development?
Absolutely, sir. So thank you very much for bringing in this question. So I would like to agree with Sirisha and Dr. Rahul Purwar. As a kid, as a college-going student, so the ambitions, aspirations are completely different. As a student in the biology, always I want to become a MBBS doctor. The goalposts continue to change. We don't have one single destiny and destination. So when it doesn't happen, then as a student, we need to move away from what is not happening and focus on something. So from my early career, I want to become a MBBS doctor. Then I moved to genetics in the undergraduate. I did masters from biotechnology. I always want to become a doctor and do a PhD. And I started doing a PhD on the cord blood stem cells. That point of time, it was Asia Cryosel and Manipal group of academy. Unfortunately, I could not continue after 2 years because of various reasons. But I never get demotivated the way that things were moving. I quickly changed my mindset and tried to transform from lab to road. So I pick up a application job where I can support scientists like you, bringing technology, instruments together and try to help you how fast you can utilize these technologies. That's how my journey started in Beckman Coulter way back in 2010. Now I'm the country leader for Beckman Coulter Life Sciences and also dual hatting into the Danaher. So Danaher is our parent company. We have group of operating companies like Cytivapol who are into biotechnology. We have diagnostic companies like Radiometer, Cepheid, Molecular Diagnostics, and Leica Biosystems. And we have a bigger platform called Danaher Life Sciences, where Beckman Coulter Life Sciences, SciX, Molecular Devices, Phenomenex, Leica Microsystems. There are a group of 10 to 12 companies that we have as a company under Danaher. We strongly believe in innovation at speed of life. Innovation can happen in two ways. One, as a industry partners that we can develop certain innovative products, and academicians like you can develop innovation. But major gap that I see in this is the awareness at the student level. Students at a student stage, when whatever I have learned in the lab are not applicable when I go for a job. The technology, the instrument, the science behind what we try to use might be useful for only 50%. When I go to industry to do a job at the lab or as a scientist, it can be completely different. So that's the challenge that I have faced personally. From Danaher, what we try to create an ecosystem where two levels. So one is foundational level where we can create an awareness, bringing students from colleges to our facility where we have got technology in one place and showcase them how the R&D will happen, how the manufacturing happens in India, how different categories of the instruments can help reach scientists' goals. That's one level of support we give. Second, we also sponsor from Danaher under the CSR initiative. Some of the postgraduate students, PhD candidates, so we sponsor their education from Danaher. That will immensely help them to pick up the pace and we will try to imbibe them in the company at one point of time. Third level that we are trying to do is the strategic level of partnerships with the industry and academia. What we currently see is the gap on the skill development, training, and resources. So these are essential for any company, even the startup company or established company. They need manpower, and for them, these technical skills, skill development, hands-on training is important. So we are partnering with renewed institutes across India and trying to build this ecosystem of developing
the
technology.
focus now in various fields is manufactured in India. Make in India.
Yes.
Look at the DRDO.
Yes.
The kind of equipment that they make now. I want to see a day when Becton Dickinson is known as the Ram Lakshman Company here, rather than Becton Dickinson. Why don't we manufacture those equipments here only? Why do we have to depend on that? I want to see if there is any question from the audience here, and then I will go back to my panel here for a second round. I don't know how much time do we have. Is there any question from the audience at this stage? So the point that I was trying to raise was, of course, Rahul said startups are facing a lot of problems. Of course, I know that. I've been associated with one startup that I told you about, and I know every day there are so many hurdles. Even though the government claims and the government says that we are trying to facilitate for these startups. Not only that they don't have funds, they work on a very limited funds. The funds is a big issue. But also there are other certifications, this certification, that certification. I think these have to be made simpler there. But our focus we have to emulate like the DRDO, start thinking in terms of manufacturing equipment in India. Any thoughts on that? Anybody?
Absolutely. Like unless you control the entire value chain in the manufacturing process, basically from the lab to the market, unless you control entire value chain, you can never be affordable. The reason we could do, especially in CAR-T, if you see the example I was telling in the talk, that because we control the IP, we own the patent, then we manufacture all the ingredients for the CAR-T cell therapy, like plasmids, lentiviral vector, CAR-T cell, and the quality control. So once we have entire thing, that is one of the reason we can bring the cost from the 4 crore to 26 lakhs. If we have, despite we use not make in India equipment, not make in India reagents, all reagents are all coming from the Danaher or some other groups. Despite that, we could only do this much. And now you imagine, as you rightly said, if we make manufactured equipment in India, even the media, reagents, other things also make in India, this cost can further come down. So make in India is great effort, absolutely. And I'm sure it will happen as well, because in India, it all depends where the market is, company always flood in that direction. So days are not far. Imagine when I applied for my first grant in government, just not too long ago, sir, in front of you. And that time it was said that these are like gestation period is too long, CAR-T cell therapy is not India's problem, right? Yeah. CAR-T cell we don't need here. We need here 5 rupees medicine, 10 rupees medicine. From that change, now you see majority of the companies are doing CAR-T research. Majority, like largest companies in India, the biggest one, they all have CAR-T programs. Major institutes have the CAR-T programs, whether it's a DBT, CSIR, or like so many scientists now are working on these technologies. So when they see the light, they are flocked there. Same thing will happen once they see there's enough media uses, Indian company will start making the media. If they see a lot of equipment, I think people will start making equipment. So things will happen. The good thing is the intention of the government, intention of the scientists is positive. Yes. People are thinking very positive and they believe that it will work.
Keep a positive view that things will happen.
Yes.
Suman, you have a lot of experience on engineering innovation in healthcare. How do you think that we should move forward to make sure that the engineering innovation can be broadened? We had in AIIMS for many years at the Department of Biomedical Engineering. And in the 70s, we all used to go and see whether biomedical engineering for what in AIIMS. But today, I think departments like those are very, very important. Your views on that?
Yeah, so I will tell 2-3 points which are coming from my experience. One important aspect of this manufacturing is design for manufacturing. And one of the big challenges is that the standard way of developing prototypes and then products that in many of the academic labs people follow are quite deviated from what is required for upscaling with certain quality standards typically needed for medical devices and diagnostic technologies. So that awareness and implementation from the design stage to all the way, starting from your first in vitro trial, then when you get into the clinical trial and then upscaling your product, all these things. Number 2 is what I would say, not much awareness on the regulatory compliance. So usually when design or manufacturing is being exercised. Typically here as a part of the medical applications in biomedical domain. There are so many aspects which are related to the regulatory issues. And if those are not considered, then towards the end when it comes to validation and certification and all, that, there is a chance that it deviates quite a bit from where it should have been targeted. The third point is that in manufacturing, it is very important to also have the supply chain and other things well established. And this is something in the medical device manufacturing arena is not so much well standardized when it comes to the MSMEs. It is quite well standardized if you go to the big giants and all. But I find it quite something where the gap has to be bridged because we work quite a lot with the MSMEs for certain joint programs for product development, etc. We find that that essential thing is missing. And my fourth point, which is actually a combination of all and possibly a recommendation, is that we will do well if every such project which has the potential of coming to that commercialization or at least field adaptation, from the very beginning the academic team is connected in an ecosystem with either a startup or an established industry. Then when they move together, they will complement their relative abilities. And in that way, it will actually get into the successful thing. I am seeing quite a few now doing like that. It's, the culture is changing. But I believe that this should be brought as a part of at least a soft mandate. I mean, if not a very hard mandate. This is what is my own impression.
If you look around, you see small countries like Israel, Holland. Every day there's a new company from there. And I have myself have the experience of interacting with them, and I see there's so much of interaction between industry and academia there. Sirisha, you are the youngest of the group over here. I want to see your dreams of how we can bring up the two of them working together hand in hand.
So, two things I just wanted to add. First thing, whatever we are talking now is not just a scientific challenge, it's an ecosystem challenge. So the solution has to be a multi-pronged approach. Everybody has to go hand in hand. The policymakers, the industry, the academia, everybody has to partner, right? And I think a couple of months back, I went to hear about PIRP, Promotion of Industrial Research in Pharma. It's a government initiative with a 5,000 crore. They're expecting, or their strong intention is for industries to set up centers of excellence within the academics, like especially NIPERS, right? So I think that's a great initiative. And even for us, even from an industry point of view, the challenge we see is the skill set is not industry ready. Sometimes they need a bridge course to get into the industry. If I can give an example to relate, right? Let's say we have some of the equipments which are high-end, let's say $1.5 million equipment. Sometimes we expect the workforce to come on board and handle it independently. One, they would not have handled independently in an academic setup. Two, sometimes industries don't have the time to give so much of training to them. That is where bridging and working together helps. Third point I want to make is when sir was actually saying that we have to go hand in hand. Early engagement is the key. Like, let's say if we can have an industry representation in a PhD DC board, right? That early engagement, industry also will understand what is the innovation that's going on, what's the product that's likely come, can we commercialize it? Because most of the innovation happens in academics. But commercialization, scaling up, or let us say clinical development, that can happen as a partnership with industries. So yes, there's a lot we can do.
Thank you. Those are very nice points.
One very small point, just
Yes.
1 second point.
Yeah.
I think failure is not rewarded in our country. At some point, we have to say to youngsters that yes, success is rewarded, but at the same time, if you fail in something, some like these kind of projects, and a startup is bound to fail. 99% will fail.
That is why I asked you in your day-to-day effort, how much is your success rate? In fact, I wanted to ask you, how much is your failure rate?
Yeah. So I always tell my students and my colleagues, there's 1 or 2 days in a year you are happy, and rest you just work hard. Right?
Come back to you, Gundu sir. Companies like yours, Becton Dickinson and all, you have very strong sales teams. You go around and you show everybody dreams and all that. My point is, when do we see big companies like you looking towards manufacturing make in India?
Sir, I think we are the early adopters of manufacturing in India, from Denher said. I'll give you 2 examples. You must have known a company called Cepheid. Cepheid manufactures gene expert technology systems and the RNA, DNA extraction systems. That's widely used for COVID testing. And we are one of the exporters of gene expert kits. And the manufacturing site is right in India, in Bangalore. The other one, today we are into immunology, and you did mention about Beckman Coulter as a flow cytometer technology. We manufacture antibodies. One is liquid reagents that everybody know, but we also develop Dura innovation. That's a dry reagent technology. So that's again back in Bangalore, and we manufacture those reagents here for both indigenous customers and also for export purpose. So I would say we are early adopters of this. We strongly believe in putting efforts in the R&D, process development, and also in the manufacturing. In coming years, you might see a lot of such initiatives from companies like Denher and investing in India and going to kickstart. So definitely we will do best.
We have to wind up. I know that there's a lecture by Dr. Bhatnagar there. I don't want to stand between, because this is a topic that we can keep talking about. I think 3 things came out very clearly from our panel here. One, academia industry interaction must be promoted and encouraged. Number 2, as I said, our students in their 2nd or 3rd year of the PhD, they should be sent out for 3 months of internship in an industry. And number 3, what you said, Sirisha, that industry should come forward to establish centers of excellence in partnership with the academia in the academic institutions there. Thank you so much. Is there any question from the audience? Just 2 I will take, because I don't want to stand. Yes, please.
Yes, sir. Sir, I was just thinking, many of the people from academia, they may be interested in, answering a question which industry experiences. But the point is, where is the information? There is no platform. There should be some national level, interface where a number of industries, express that these kind of problems we face, so that industry people, so that academia people can start talking to them. There's the huge gap. How to fill that? That should be,
Good suggestion. We will keep talking about it during dinner time as well.
Yes.
Anybody else having any point? Yes, please.
Sir, not the question, but just a small suggestion. As IITs, all these companies, they go and have the campus interviews for the selection. They should come to academia so that our PhD students, our MSc students, they should know where are the opportunities. They are looking for that.
We do come for both internships and placements.
Well, with this, okay, the last one, please. Yeah.
So I've been actually interacting with few startups, MSME startups. So they were saying that at the regulatory level, they face a lot of issues. Though the owners are not actually from biology background. They are from some other, but they have the business mind. So they work in life sciences. They are having good products because they get it tested from us. But they feel that taking it ahead, they face a lot of problems in regulatory issues at the whatever DCG or whichever regulatory bodies. So that's why they face, if that gets smoothened, probably more and more people will come into startups.
Thank you. Although the organizers have not come to tap my back and said your time is up, but I think our time is up. I know that our time is up. So thank you so much.
Thank you very much.
I want to thank all my panelists here and thank you all for your attention. Thank you so much.
Hello. It's okay, sir. Your talk was too great to be stopped. Thank you, Professor Mehra, and all our distinguished panelists for an engaging and insightful interaction. As a gesture of our gratitude, may I request Professor N.K. Mehra, sir, to kindly present the mementos to our esteemed panelists, to Professor Suman Chakravarti, whose remarkable contribution and leadership continue to inspire the scientific community. To Professor Rahul Purwar, whose pioneering work in cell and gene therapy has opened new pathways for cutting-edge healthcare in India. To Mr. Chaitanya Gundu for his commitment to advancing innovation in the life science industry. And to Dr. Sirisha Potala for her impactful leadership in global pharmaceutical strategy. And now, as a mark of our gratitude, may I invite Dr. Sameer Srivastava, sir, to kindly present a memento to our moderator, Professor N.K. Mehra. We extend our heartfelt thanks to Professor N.K. Mehra, sir, for his insightful guidance and gracious moderation of this session. Thank you, sir. As we conclude this session, let us take a moment to speak about Indian Immunology Society. The Indian Immunology Society was established in 1972, stands as one of the premier scientific communities in the country, dedicating to advancing the field of immunology. With a vibrant network of researchers, clinicians, and academicians, IS promotes cutting-edge research, capacity building, and knowledge exchange across India. Through national conferences, workshops, collaborative platforms, and student engagement initiatives, the Society continues to nurture scientific excellence and strengthen India's contribution to global immunological research. As we move ahead, we now come to another special segment of our program, the felicitation ceremony. This segment has been allotted 20 minutes, and while distinguished contributions such as his can hardly be confined within any duration, we humbly request you to kindly cooperate. May I now invite our distinguished chairperson for this segment, Professor Amit Awasthi, sir, President of Indian Immunology Society, and Dr. Kaustav Nayak, sir, Secretary of Indian Immunology Society, to kindly come forward and lead the felicitation ceremony.
Thank you. So this is the last session, I believe, of the day, and this is a special session, and this is an honor for all of us to recognize the contribution in Indian science and research, education, you name it, as a distinguished scientist honor or award to Professor Rakesh Bhatnagar, We all know Professor Bhatnagar, and I will not really spend too much time, but I request Professor Santosh Kar to come on the stage and introduce Professor Bhatnagar for this talk, and which will be followed by the felicitation. Thank you.
Thank you. We are already 1.5 hours late, and it is a pleasure to finish this today's conference, with this address by Professor Rakesh Bhatnagar. Thank you, Amit and Kustub, for requesting me to introduce him. We were colleagues at JNU, and I have a written biodata, which I will not completely read because it will take time, but I would just like to mention that it gives me great pleasure today to introduce Professor Bhatnagar to all of you because of a very special reason. 1982, I had just joined Professor Talwar's National Institute of Immunology at AIIMS, and I was asked to organize an immunology teaching program. Please remember, 1982, 18 years plus 25 years, 43 years ago, I had started teaching immunology to students in India, following Professor Talwar's footsteps. And Rakesh and his wife, Nirupama, she was also a scientist. She became a distinguished scientist in ICGB and retired life. They both are leading. When she is leading, Rakesh is a very active person. So 1982 immunology, मैंने Professor Bhatnagar को पढ़ाया था immunology, and today he is being awarded the distinguished award as an immunologist. So partly credit goes to me as a teacher. Professor Rakesh Bhatnagar is a distinguished scientist, visionary, institution builder, because he has been Vice Chancellor in Banaras Hindu University and 2 other universities, Amity, as well as another university, and one of the India's most respected academician. He's a National Professor of Biology located at JNU. He has more than 200 publications, 6,000 citations, and I know, because I worked with him for almost 25 years. When I retired in 2011, I joined, about a year after he joined JNU, both joined as associate professor, but he became professor, I also became professor. We used to compete and he was taking care of molecular biology. I was teaching immunology in JNU. And we had many good occasions to be friendly, and also some small occasions where he used to fight. It is my great pleasure today to introduce you to a visionary and who has been continuously devoting all his time in research and teaching. And his major contribution was recognized by this anthrax vaccine thing that he made. And I'm not going to go into it because I am a habit of speaking too much. And if you give me the time, I will speak for next 1 hour. I'm not going to do that. Only thing is that, Rakesh, it's a great pleasure for me to invite you to come and give a little glimpse of what is going on in your mind about immunology, biology teaching, because you are National Biology Professor. What is the future of our science in India today? Rakesh,
Thank you, Santosh, for giving nice words for me. But today's topic which I have been given is to summarize what all I did during the last three and a half decades back in JNU. So I will be talking about, can I get it on the screen? Okay. Good evening, everyone, but I'm sure you all would have been tired by now, so I'm not going to increase your boredom, and I will not stand between you and your dinner. So I joined Jawaharlal Nehru University in almost end of 80s, and we started teaching biotechnology because the country thought that if the biotechnology industry will come one day, where will we get manpower? So Department of Biotechnology and UGC decided to float some courses in the universities, and JNU took the lead, and we started teaching biotechnology to our students. And as Santosh told, that half a dozen of us were imported from United States who were practicing biotechnologists to come and share our experiences with the younger minds and train them. Fortunately today, all of them are doing well. I think this morning I met few of them here who are holding very high positions. Many of them are owning biotech industries in United States and in Europe. Many of them are presidents, vice presidents, CEOs of the companies. So that is where me and Santosh started. He used to take care of immunology, as he told, and I used to take care of molecular biology. But my interest was there in developing the vaccines, because our neighboring institute, National Institute of Immunology, founded by Professor Talwar, gave a lot of inspiration to all of us to develop vaccines. So I still remember in early 90s, there was anthrax epidemic in West Bengal, and country didn't know what to do. So I went to Department of Biotechnology to ask if there is a possibility of getting some funding to develop recombinant vaccine against anthrax. So they said, quickly give us. I said, how quickly are you going to give me the grant? We have a parliament question, so we can answer that we are in a process of funding a grant. So that's how I got my first grant from DBT and started working on developing recombinant vaccine against anthrax. I think anthrax is very well written in textbooks of microbiology, immunology, everybody has read that, so I am not going to get into those details. So that is how Bacillus anthracis looks under the microscope. It has two plasmids, one we call it PXO1, another we call it PXO2. PXO1 encodes for protective antigen, lethal factor, edema factor. These are all virulence factors of anthrax. And the PXO2 encodes for polyglutamic acid capsule. Mind it, it is D-glutamic acid capsule, it's not L. So therefore, no enzyme will come and clip it. So it gives a protective covering to the bacteria to survive in the host, and the spore becomes vegetative cells, and they start making protective antigen, lethal factor, and edema factor. So it makes these are the, some of the anthrax epidemics which happen in India, in Jharkhand, West Bengal, Odisha, Andhra Pradesh. Even in IIT Madras, I think a couple of years back, they had anthrax because they have a lot of forest area within IIT Madras. Even in neighboring countries like Thailand, last year we had anthrax. So anthrax is of three kinds. One is cutaneous anthrax. As the name indicates, it's a skin anthrax. So what happens that if you have little cut or abrasion in your skin, a spore of Bacillus anthracis can come and get inoculated in your skin. It becomes little pink, then it turns to vesicle, and eventually it becomes a black scar. It doesn't bother you too much, so you tend to ignore it. But if it is not treated, I think 1% people only, they die. However, the gastrointestinal anthrax is more dangerous, and it is very common in our tribal areas where, you know, when they see an animal, big animal died in the forest, they think it's a gift from God. They try to take the meat and sometimes try to roast incompletely, or sometimes they don't cook it properly, and they get intestinal anthrax. So they get pain in abdomen, and 50% people die if you don't treat them immediately with the antibiotics. The most dangerous kind of anthrax is inhalation anthrax. If you inhale spores of Bacillus anthracis, which you know, they look like dust particles, they go to your lungs and they try to grow in alveolar macrophages. They make a lot of toxin, which eventually gives you a lot of fever, difficulty in breathing, and eventually the person dies if the person is not treated immediately with the antibiotics. But let me ask you, when do you take antibiotics without seeing the symptoms? And symptoms take about 2 to 5 days to appear. So 99% of the people, even if they are given antibiotics for 60 days as per WHO recommendation, they die. So therefore, it is very important to develop vaccine against anthrax. To understand my talk, if you understand these 3 molecules, it will help you. Protective antigen is the binding molecule to the receptor. It is a 83 kilodalton protein. Lethal factor and edema factor all by themselves are not toxic, although the name is dangerous. They only become toxic when they are combined with protective antigen. Okay? Edema factor is calmodulin dependent adenylyl cyclase, whereas lethal factor is a zinc dependent protease which can clip MAP kinase series of enzymes. Let me show you little cartoons to understand how this disease progresses. Protective antigen goes and binds to eukaryotic cell surface receptors. All our cells have these receptors. So they come and bind to protective antigen, and a protease sitting, which is furin, it comes and clips the protective antigen to create a binding site for lethal factor or edema factor. In fact, they both try to compete to bind the same site on the receptor. And then they go inside by receptor-mediated endocytosis, and this endosome is acidified by pumping of the protons, and eventually they come out of the endosome and bring out their cytopathic effects. If you remember 2001, okay, a notorious guy in the United States working in Frederick, where I worked in that lab, this guy has made some anthrax spores and tried to send it to many people in media and congressmen. His theory was, if you want to increase the funding of anthrax, it takes only few envelopes. And that's what he did. So when 13 people got infected and 5 of them died, the FBI was given this responsibility to find out where do these spores come from. And once they went to the lab and talked to everyone, the day his number came, his name was Bruce Ivins, he committed suicide. You can understand. Although they could not prove it beyond point, but why should somebody commit a suicide unless he has done this job? So what is the vaccine situation today? Till date, animals are getting live vaccine, live spore vaccine, which is also even made in India, in Hyderabad. Russia is still using animal vaccine. USA and UK, they grow Bacillus anthracis strain which is devoid of PXO2. It cannot cause disease, but it makes protective antigen, lethal factor and edema factor. So they take the supernatant of this in the fermenter and try to precipitate with alumina and take it as a crude vaccine, which they shoot it to their soldiers. And this technology to make this vaccine is tightly guarded by US Army and UK Army. They don't share it with anyone with the reason that this can be used as a biological warfare agent and terrorists can shoot themselves with this vaccine and then throw the anthrax on anyone they like, okay? So therefore, if anybody has to develop this vaccine, it has to be an indigenous effort. So that's what we did. We wanted to genetically engineer vaccine against anthrax, so what we did, we took out PXO1 plasmid from Bacillus anthracis and just do a PCR amplification of the gene and put it in an expression vector and grown in and try to see whether it makes protective antigen or not. And when we ran the gel, it makes a good amount of protective antigen. So what we did, by following few steps of purification, we purified the protective antigen and tried to see whether it behaves like native protective antigen or not. For example, I showed you in the cartoon that it goes and binds to the protective antigen to the receptor. So we wanted to see whether it is able to bind to the receptor. So what we did, we took native protective antigen as well as recombinant protective antigen and radioiodinated it and tried to see whether it can bind to the receptor or not. It has the same capability of binding the receptor. You see about 80,000 counts in both the cases. Then we wanted to see whether protease sitting on the cell surface is able to clip it or not. It was able to clip from 83 kilodalton to 63 kilodalton. We wanted to show whether it is able to bind to the protective antigen or not. We could show it on the native gel, it was able to bind. Then we wanted to see whether it is able to kill the eukaryotic cells or not. So protective antigen made by recombinant route as well as lethal factor, if they put together, it is able to kill the macrophages in a dose-dependent manner. Whether we use native antigen or recombinant antigen, the graphs are almost superimposable. So we made the right molecule. Then we wanted to test in the mouse model whether it is able to kill, whether it is able to protect the mouse from the infection. So what we did, we immunized the mouse. Mouse is a very easy model. Only few spores can kill the mouse model. So we tried to inject them with this recombinant vaccine and then eventually tried to challenge them with the live virulent anthrax bacteria. And we could see about 66% protection in the animal model. And as COVID has taught all of us that even if you are getting 50% protection, you should go ahead. So then we did the experiments on guinea pigs and followed by rabbits and on monkey model, and eventually we could show that in monkey model it is able to protect even 100% monkeys by giving the live challenge. Then at this point of time a company contacted us. They wanted to buy this clone. So we said, yes, we can give you the clone. What is JNU getting out of it? So they said, we will give you 40 lakhs of rupees in few installments. So once we take the clone, we will give you 4 lakhs of rupees, and then we will upscale in the company and see whether it can make a good amount of vaccine or not. I asked them, what if it doesn't make that kind of levels? We will return your clone. So I asked them, what about if you just streak my clone and come in the market after 2 years and say we have in-house developed? And I don't have financial muscles to take you to the court because you are a drug company, I am a poor professor. So I went back to my funding agency and asked them, why don't you fund me upscaling also? And they were very nice, DBT. They funded upscaling of the recombinant vaccine production. So that's what we did. and we made up to 1 million doses in a 5-liter fermenter. So that when we talk to the company, we can say, hey, we can make 1 million doses, are you ready to buy? So that's what we did. Then another company came and they were very bullish and they said, yes, we are ready to buy. They asked me, how much money do you want? I'm talking about 2001. So I asked for 1 crore, which is, something like 20 crore today. So the company agreed immediately, yes, we can give you 1 crore. I thought maybe I asked too little. So what to do now? So I said, I'm giving you this clone if you are only exploiting Indian market. But if you want to exploit international market, you have to pay 2 crores. The company agreed. We don't want a competitor, you give us the whole thing, whole rights. So I gave them, and then they went for preclinical toxicity, then animal efficacy trials, then followed by phase 1, phase 2, etc. clinical trials, and they completed that. And that gave us a lot of patents and lot of name and fame, as Santosh put it. But let me tell you, science never ends. Protective antigen is a good immunogen. But if you add little lethal factor and little edema factor, it becomes a better immunogen because it mimics the disease more close to the real situation. But you cannot add that because the moment you add, it makes a toxin. And you don't want to add. So we decided, why don't we make a lot of mutations where my knowledge about molecular biology comes, and try to make mutations which are not able to connect to each other so that they will never make a binary toxin. So that's what we did. So we made a lot of mutations in protective antigen, in lethal factor, in edema factor, and finally made them non-toxic so that they can be added in the vaccine preparation to improve the vaccine. This is what all we did. Considering the time constraint, I am going a little faster. Then we wanted to develop DNA vaccine. So what we did, we took protective antigen, put a lot of sequences upstream and downstream so as to direct the antigen at the right location in the cell. And we could see that if we put LAMP and TPA sequence, we are able to get about 88% protection in mouse model. Protective antigen is a long molecule, 735 amino acids. So we wanted to see whether we can find out what are the immunogenic domains which are really responsible. So what we did, by the help of bioinformatics, we tried to make certain peptides which could mimic almost the same kind of immune response. Then we went for developing, because it's an animal disease, so we wanted to develop some edible vaccine. So we cloned the protective antigen in tobacco and then finally in tomato, and nowadays we are doing in sorghum to make the vaccine available for cattle also. Now the next question was that what if somebody has not taken the vaccine? So you should have a very good post-exposure therapy. So what we did to create the post-exposure therapy, we targeted 3 things. 1, a toxin, antitoxin mediated therapy. 2nd, novel inhibitors, natural as well as synthetic, as well as therapeutic antibodies. So this was the toxin, antitoxin, and we tried to find out how to block this interaction so that the cells can be killed all by itself. So we made certain peptides, but they were not very effective. Only 40% inhibition we could get. Then we wanted some natural molecules which will sit in between lethal factor and protective antigen and will not allow their interaction. So this QDH as well as this TGG, we tried to create, and it gives about 80% protection in cellular model. We have yet to do the animal experiments. The third and the most important achievement which we had was to develop therapeutic antibody. So what we did, we I think I will straight away go. Suppose you try to, LF and EF, they bind to the same place in protective antigen. So if you try to raise the antibodies against this binding region, you can block both the molecules. And that's what we did. See, here is lethal factor and edema factor. We cloned sequence and we found the sequence which was exactly the same. And we raised antibodies against that. And we found 2 antibodies which were able to neutralize lethal factor as well as edema factor. See, edema factor, we put it in the mouse model, and by this antibody we could neutralize it. Sorry. And when we use the antibody in a live challenge model, we could get 60% protection, but when we did it with the antibiotics, we could get 100% protection. And now we have humanized this antibody, and we have filed the patent in April 2025, and gradually we were looking for some company which can take it further for preclinical and clinical studies. Thank you. So thanks to funding agencies, my collaborators, and the most important, PhD students who worked day and night. They slog, we only talk. And these are some of the recognitions I got from ICMR, from President of India, to develop this all. Thank you. This is my lab, which has a BSL-3 laboratory, and on top my research lab is there. Thank you.
Thank you, sir.
Some of my students.
Thank you, sir. Thank you, Professor Bhatnagar, for your the overall journey about the anthrax vaccine. And I'm very sure that all young minds will definitely take the inspiration to develop some more such product for India. Since this is a award session, so therefore there is no question answer. But in case if somebody like to ask any question to Professor Bhatnagar, you can always reach to him through the email, because he will be moving back tonight. So with this, we will felicitate Professor Bhatnagar, and I request the organizing secretary, Professor Ambak Rai, to come on the stage for this ceremony.
Thank you.
Thank you, sir, for graciously leading this wonderful felicitation. Thank you. As we move forward, it's time to celebrate another dimension of our gathering, culture. Scientific excellence may enlighten our mind, but culture is what connects our heart, reminding us of our heritage, rhythm, and creativity that shape us who we are. To take us into this vibrant cultural segment, we ask you to be with us for just a brief moment longer, as something special awaits. Please stay tuned with us for the next few minutes. Your presence will make this moment complete.
इधर से नहीं, इधर से दीजिए, इधर से, इधर से। ना ठीक है। हेलो हेलो। कहाँ शुरू किया था? साउंड से थोड़ी है? नहीं नहीं। भैया यार जूता उतार के आओ। हेलो हेलो हेलो।
Hello, hello.
आप ना अपना माइक दे दो। हाँ उनको बोल दो कि फैंटम नहीं चल पाएगा, आप अपना माइक दे दो। बोल दो पहले म्यूट कर दे, उनको बोलिए म्यूट कर दे इसको। मिनट दे देंगे। थोड़ा कम कर दीजिए, थोड़ा कम। ठीक है, ठीक है। ये वाला। हाँ ठीक है, थोड़ा बढ़ाइए।
हेलो हेलो
배달에 가다 기나 다 다다 기나 다 किटे घिना ना
वो इनका है, वो ये आ रहा है। सारंगी चेक करिए।
हेलो
मुझे गाना का जैक का प्रॉब्लम है। वोकल का जैक का प्रॉब्लम है कुछ। हाँ? नहीं नहीं, एक्चुअली इनके वोकल के माइक
हेलो 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 हेलो
हेलो हेलो
इसमें बता दीजिए।
हो गया हो गया
इसको चेक कर लीजिए।
Hello, hello.
Banaras, the city where rhythm flows like Ganga and the art breathes in every corner. In its very air lives a dance form that mirrors the spirit of the city itself, Kathak. Just as Ganga flows with devotion, Kathak moves with grace. Each step echoing like temple bells, each spin unfolding a sacred story. Through its expressive gesture and soulful rhythm, Kathak in Banaras becomes more than an art. It becomes worship in motion. In Banaras, dance does not merely exist. It lives, breathes, and blesses all who witness it. And from this divine soil emerged a true legend, Pandit Rudra Shankar Mishraji. A torchbearer of the Banaras Gharana, his every step reflected discipline, his every expression carried emotion, and his every performance became a soulful conversation between the divine and the dancer. Pandit Rudra Shankarji devoted his life to preserving the purity of Kathak, bending traditions with grace and spirituality. His abhinay spoke louder than words, his rhythm mirrored the heartbeats of Banaras, and his dedication turned every stage into a temple of art. And now, to let our hearts dance with the rhythm and devotion, we invite on stage the living rhythm of Banaras himself, Pandit Rudra Shankarji. Let's welcome him with a huge round of applause.
शीश गंग भाल चंद्र नेत्र लाल कंठ व्याल भस्म युक्त नग्न तन जटा जूट धारी हैं भूतेश्वर नागेश्वर डमरू त्रिशूल धर असुचि का घर बैल जिसकी सवारी है सत्यम शिवम सुंदरम कहे जिसे वेद कन्या का तुम्हारी वही शिव अधिकारी है ओम नमः पार्वती पते हर हर महादेव गिरि कैलाश शिखर पर नाच धमक धमक ध्वनि मृदंग बाज शीश गंग अर्ध अंग विराज उमा रमा हरि सब सुर साज कर त्रिशूल डमरूले नाच शूजम दम दम छन न न न न न नट धैय धैय दा धैय धैय दा धैय धैय दा शूजम दम दम छन न न न न न नट धैय धैय दा धैय धैय दा धैय धैय दा शूजम दम दम छन न न न न नट धैय धैय दा धैय धैय दा धैय धैय दा
करुणा कर कर हरे जय कैलासी जय अविनाशी जय कैलासी जय अविनाशी सुख राशि दुख जय शिव शंकर जय करुणा कर कर ओम हरि ओम हरि ओम हरि
जल प्रभाव पावितस्तरे कलेवलं विलंबित भुजंग तुंग मालिक टमट 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 निनाद भट्ट मरव चकार चंद तांडवम तनोतु न शिव शिव तटा कटा हस प्रम प्रमत्तलिंप निर्जरि विलोल वीच वल्लरि विराजमान मुरधनि तकत 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 चलल्ललाट पट्ट पावन किशोर चंद्रशेखर रति प्रतीक्षणम मम रति प्रतीक्षणम मम रति प्रतीक्षणम मम
Namaskar and pranam to everyone present here. It's an honor for me to perform the North Indian classical dance Kathak in front of you. ये बहुत सौभाग्य है कि मेरा ये दूसरा बार है यहाँ पर और मुझे बहुत अच्छा लगता है जब भी मैं यहाँ आता हूँ. दर्शक, the members, जितने भी लोग हैं, really feel so honored to be in between you. तो आज बनारस से आए हैं भगवान शिव की नगरी से और शिव जी की आराधना कर चुके हैं. थोड़ी देर हम अपने कथक की ओर चल रहे हैं जो पारंपरिक है लेकिन बहुत ही सुंदर तरीका है मैथ्स को समझने का, बीट्स को समझने का और इमोशंस को समझने का. तो आपके समक्ष थोड़ी देर हम उस कथक की प्रस्तुति कर रहे हैं जिसको रामायण काल में लवकुश जी ने जिसकी शुरुआत की थी, कथावाचन शैली, और वो शैली आज वृहद रूप लेते हुए नृत्य का स्वरूप ले चुका है. तो थोड़ी देर आपके समक्ष हम प्रस्तुत कर रहे हैं एक ताल, 3 ताल. अब खास बात यह है कि ये जितने भी आप यहाँ संगत कलाकार देख रहे हैं ये गति देते हैं मुझे. 16 मात्रा है, 3 ताल, 16. जैसे हम कहते हैं कि कृष्ण की 16 गोपियां हैं और एक कृष्ण है, तो वो सारी गोपियां कृष्ण को पाना चाहती हैं. वैसे ही हमारा डेस्टिनी 1 है, वहां पहुंचने की कोशिश करते हैं. तो थोड़ी देर 3 ताल आपके समक्ष प्रस्तुत करते हैं. धन्यवाद. अभी behind the stage मैं पूछ रहा था कि is it the conference on the immune and technology? So she said yes, sir. We are collaborating and doing some work. तो हमें दौड़ने के लिए, हमें चलने के लिए, बात करने के लिए इम्यून की बहुत ज़रूरत होती है. तो ऐसे ही एक जानवर, पशु कह सकते हैं, घोड़ा है, इसकी इम्यून बड़ी अच्छी थी क्योंकि दौड़ता बड़ा तेज है वो. तो उसके दौड़ने की जो आवाज़ है वो कथक के पैरों से कैसे आती है, वो मैं कोशिश करता हूँ. When I dance Kathak, so दृष्टि थोड़ी अलग होती है हर चीज देखने की. So I was just seeing that this is the 52nd conference जो यहाँ पर अभी हो रहा है. So I'm trying to present opposite of it. मैं 25 चक्कर की कोशिश कर रहा हूँ. That's the opposite of 52. 52 is quite tough, not tough, but अभी माइंड में 25 मैं पीछे बैठकर बना रहा था. तो it just came out. और चक्कर की खूबसूरती ये है कि ये सिर्फ बाएं पैर पे होता है. राइट से काउंट करते हैं हम. 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 1 2 3 4 5 25 चक्कर.
One, two, three, four, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 1, 2, 3, 4, 5. हम अक्सर बात ये करते हैं कि the result that we got, but the process is also important. हम शुरुआत करते हैं बहुत छोटे से, from play group, nursery, और अच्छे पोस्ट पे वर्क करते हैं. और उस दिन हमें खुशी मिलती है कि नाउ आई एम एट दिस पोस्ट, ये मेरा ड्रीम जॉब था. बट उस वक्त जब आप उस प्रोसेस को सोचते हैं तो आप सोचेंगे आपने कितने साल दिए. तो वैसे ये कॉम्पोजिशन है जिसमें वो पूरा प्रोसेस दिखता है
और एंड में वो धा जो हमारा डेस्टिनी है वो आता है, जहां हमें जाना है। तो किस प्रकार से है? धा दिन का दिन नग तेरे गिट तक 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 ता 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 धा गिट तेरे धा 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 धा। बेस ज्यादा है इनके में, जो बोल रहे हैं उनका बेस थोड़ा कम कर दीजिए। We have languages to talk, to conversate. We have WhatsApp, Facebook, Insta, LinkedIn. अभी बहुत सारे बंद हो गए, कुछ चल रहे हैं। तो यहाँ भाषा की जरूरत पड़ती है ना, इंग्लिश या हिंदी, कभी इंग्लिश, कुछ भी। सो क्रिएटिविटी यहाँ भी है, और इस क्रिएटिविटी का नाम है कि हम भाषा का प्रयोग नहीं करते, आवाज़ों से बातचीत करते हैं। तो घुंघरू की क्या आवाज़ है? वो तबला सुनता है और उसका जवाब देता है। तो कैसे वो बातचीत देगी? एक ऐसी मैं प्रयास कर रहा हूँ। आई नो कि आप सुबह से बैठे हो एंड यू आर हंग्री एंड सम ऑल्सो आर एंग्री, तो वो हैंग्री हो रहा है। सो मैं ज़्यादा वेट नहीं कराऊंगा, आपको बस थोड़ा शांत करना चाहूँगा। तो मैं एक बारिश की कोशिश कर रहा हूँ, जिसे आप फील करो तो शायद आपको बारिश का एहसास आए।
इसी बीच में जो हवा का झोंका हम लोग के कानों के पास से गुजरता है, वो जो बारिश का बौछार होता है, उसका आवाज़, वो घुंघरुओं से।
एक बहुत ही खूबसूरत चीज़ है जहाँ पे हम कथक को देखते हैं, आवाज़ बहुत पावरफुल, एक सॉफ्ट चीज़ भी है, बहुत सारी चीज़ें हैं। तो जैसे हम आजकल क्या बहुत पहले से है, चलने का अंदाज से हम पता लगाते हैं बहुत सारी चीज़ें। तो कथक में भी चलने का एक अंदाज है, तो एक छोटा सा प्रस्तुत कर रहा हूँ। पिकॉक, मोर जब नृत्य करता है तो उसके पंख किस प्रकार खुलते हैं, बारिश होती है तो कैसा है, और दो चार चीजें और हैं, उसके बाद हम समअप करेंगे।
दूसरा ख्याल,
चीज़ें बहुत सारी हैं, समय को ध्यान में रखते हुए हम अपने आखिरी प्रस्तुति की तरफ बढ़ रहे हैं। कहते हैं कि वो आरती की लव है और झूमता मसान है, वो धरा की धमनियों में बह रहा तूफान है, वो राग है, रंग है, फाग है, भंग है, वो साम दाम दंड प्रचंड है, वो दृश्य भी है, दृष्टि भी है और घोर अंधकार है, उसकी आंखों के तले प्रलय भी बाहर है, वो पोर पोर रक्त, वो सांस सांस शंखनाद है, वो आरती की लौ में है और झूमता मसान है। प्रस्तुत है मसान की होली, शिव की होली।
भूत पिचास बटोरी, हाँ भूत पिचास बटोरी, दिगंबर खेले मसाने में होली, खेले मसाने में होली, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर खेले मसाने में होली, खेले मसाने में होली, दिगंबर खेले मसाने में होली, गोप न गोपी श्याम न।
होली क्यों अलग है कि ना गोप है, ना गोपी है, ना श्याम है, ना राधा है, और कौन है? भूत पिशाच है। तो होली तो अलग होनी है।
गोप न गोपी श्याम न राधा, गोप न गोपी श्याम न राधा, ना कोई रोक न कोनो बाधा, ना कोई रोक न कोनो बाधा, ना सजने ना गोरी, ना साजने ना गोरी, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर खेले मसाने में होली, नाचत गावत डमरोदारी, नाचत गावत डमरोदारी, नाचत गावत डमरोदारी, नाचत गावत डमरोदारी, छोड़े सर्प गले पिचकारी, छोड़े सर्प गले पिचकारी, पी के पेट ढपोरी, हाँ पी के पेट ढपोरी, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर खेले मसाने में होली, होली खेले मसाने में होली, दिगंबर मसाने में खेले मसाने में होली, दिगंबर मसाने में खेले मसाने में होली।
एक आखिरी प्रस्तुति जो मैं पिछले बार भी आया था और किया था, और उसी से ही समाप्त करेंगे क्योंकि वो विशेषता है हमारे घराने की। एक नृत्य जो ना हवाओं में होती है और ना जमीन पर होता है, एक बीच में ऑब्जेक्ट होता है, वो आप खुद देखिए क्या है। थाली। Thank you again for being here. For us, you all are here, it really means a lot. And also, my soul, my accompanist on Bol Padant in Manjira, Pandit Mata Prasad Mishra ji. He is my guru, father. On tabla, Shri Uday Shankar Mishra. On vocal, Shri Shakti Mishra. And on sarangi, Shri Om Sahai Mishra. Thank you so much, MNNIT, to all the teachers, to all the members, everyone behind the stage, in front. Thank you so much. It's always been a pleasure for me to come in front of. Thank you.
I think we should all stand up and give the standing ovation to this wonderful performance to this Kathak. I think it's amazing what you have performed. All the people here, please come on the stage. Thank you so much for coming here and entertaining us. at MNNIT. I hope you will come again. Sure, right? Just one question. Are you all brothers because Mishra? Okay. Okay. And this is the more real family. This is more real family.
आई थिंक आज का जो ये प्रोग्राम था, दो शब्दों में कहा जा सकता, फैंटास्टिक, फैंटास्टिक। ऐसा कथक हमने कभी देखा नहीं है, और बनारस घराने का तो पहली बार देखा है। ये तो कमाल का था। और आपकी जो कोऑर्डिनेशन है पैरों की, जो आपने मोर का डांस दिखाया, जो आपने घोड़े की चाप दिखाई, बनारस की होली दिखा दी, और फिर बाद में थाली में, ये तो कमाल चीज़ें थी, कभी हमने देखी नहीं है। तो प्लीज़ गिव हिम ए बिग हैंड, ए रियल स्टैंडिंग ओवेशन, द होल टीम, रिमार्केबल। हमारे बीच में डॉक्टर तलवार हैं, ये अभी 100th ईयर में आ गए हैं, और ये आर्ट के इतने शौकीन होते। हमने इनको देखा है शुरू से ही। साइंस में सारा दिन रहेंगे, लेकिन जब शाम के टाइम में कल्चरल का टाइम आ गया तो कहीं नहीं जाते, बिल्कुल टकटकी लगा के देगा। आज भी यही कर रहे थे। मैं इनको भी माइक देना चाहता हूँ। 2 शब्द आप कहना चाहेंगे सर? अभी नहीं कहना चाहेंगे, लेकिन इन्होंने आपके डांस को इतना एडमायर किया है। मैं वहाँ से देख रहा था इनको कि टकटकी लगा के देख रहे थे। एंड वी हैव सीन हिम लाइक दिस एवरी ईयर आफ्टर ईयर। ही इज सो फॉन्ड ऑफ दिस। और ये जब हिला रहे थे ऐसे हाथ, अपना सर हिला रहे थे, तो ये आपके लिए बहुत बड़ा सर्टिफिकेट है। थैंक यू सो मच, थैंक यू सो मच।
It was truly a mesmerizing performance by Pandit Rudra Shankarji and his team. I would now request Professor R.S. Verma sir, Director of MNNIT, and Professor N.K. Mehra to kindly present a memento as a gratitude and love. with that, we conclude an inspiring first day of IMMUNOCON 2025. Thank you for your energy, curiosity, and the passion you brought into every session. As the light dim on today's discussion, we invite you to return tomorrow for another day of breakthrough ideas and unforgettable conversations. Before we all disperse, we warmly invite all delegates to join us for dinner, which is now being served in the dining area. Please enjoy the evening, relax, and connect with fellow participants over a delightful meal. Rest well. Day 2 awaits, and it promises even more brilliance. Thank you.